if you're lucky enough to be born in a rich country in the modern world, you've got about a 0.5% chance of not making your first birthday because, you might be born with some congenital problem or genetic issue. But if you do make it through that sort of first year of life, your risk of death carries on decreasing until you get to the age of 10. And modern 10-year-olds have an incredible title. They are the safest humans in the history of our species. They've got a less than 1 in 10,000 chance of not making their 11th birthday.
Welcome to the Proof Podcast, a space for science-based conversation exploring the health and longevity benefits that come with mastering nutrition, physical exercise, mindfulness, recovery, sleep, and alignment. Facts, nuance, and trustworthy recommendations, minus the hyperbole. In today's conversation, I sit down with Andrew Steele, PhD. Andrew is a scientist, writer, and campaigner based in Berlin, Germany. He's the author of Ageless, The New Science of Getting Older Without Getting Old. After obtaining a PhD in physics from the University of Oxford, Andrew decided that aging was the single most important scientific challenge of our time and switched fields to computational biology. In this conversation, we build on learnings from previous episodes related to aging biology, discuss upcoming innovations such as senolytics and gene therapy that may help us stay healthier for longer, and revisit some of the things that we can do with our lifestyle to improve our health span. Please enjoy. You did your PhD in physics at the University of Oxford, I believe, and then, from what I understand, you had a sort of little bit of a career change and started to direct your attention to human biology and then more specifically the biology of aging. What inspired this career path for yours so far?
Well, I guess it's probably as a result of having done that PhD in physics that I was able to change career because of a graph, essentially. And I think it's actually worth talking through that graph because really, firstly, it is what inspired me to change career. But secondly, I think it's a really good way to understand the whole subject of this podcast, understand the aging process. So the graph that inspired me it's a very simple graph, actually. It's a graph of along the bottom is how old you are, and up the side is your chance of death in that particular year of your life. And obviously, all of us know that older people are more likely to die, but just how much really shocked me. So to give a few numerical examples, let's go through from the beginning of your life. If you're lucky enough to be born in a rich country in the modern world, you've got about a 0.5% chance of not making your first birthday because, you might be born with some congenital problem or genetic issue. But if you do make it through that sort of first year of life, your risk of death carries on decreasing until you get to the age of 10. And modern 10-year-olds have an incredible title. They are the safest humans in the history of our species. They've got a less than 1 in 10,000 chance of not making their 11th birthday. But unfortunately, things are all basically downhill from the age of 10. So by the time you get to my sort of ripe old age of 37, my odds of death this year are about 1 in 1,000. And it's worth just thinking about that for a moment, because actually, I quite like those odds. If that were to continue for the rest of my life, I'd live into my 1030s on average. But obviously that doesn't continue. And what actually happens is that when you become an adult, your risk of death roughly doubles every 8 years as a human being. And that means it starts out, as very, very low numbers, 1 in 10,000 at the age of 10. But you carry on doubling and doubling and doubling that number. Eventually, it can get very big very quickly. So if I'm lucky enough to live into my 90s, but unlucky enough that we haven't worked out any new medical technology in the intervening time, my risk of death every year will be about 1 in 6. So that's life and death at the roll of a dice. And, you can look at this as a human being and think, well, that's a little bit terrifying. I've got this sort of exponential wall of mortality coming at me. Or you can look at it as a physicist and think, well, what is it that's causing this incredibly synchronized increase in the risk of not just death, but all of these chronic diseases, of frailty, of, incontinence, impotence, dementia, all of these different things increase exponentially with age. And so perhaps if we could understand the biology of this process, maybe we could do something about all of these, leading killers of the modern world.
And so that's where the field of longevity science and understanding the biology of aging comes in. I believe that you listened to my episode with Matt Caberlin on aging.
Yeah, I did. I'm a big Matt fan.
From a few months ago. And at the beginning of that episode, I asked Matt how he sort of defines aging. And from memory, he sort of spoke about the difference between chronological and biological aging. So I believe what you were just explaining there is that there's that exponential increase in our risk of dying as our chronological age goes up. And So Matt spoke to the difference between, I guess, chronological age and biological age, and sort of why he likes to focus on biological aging, what's happening at a sort of cellular level as an organism when you go from being young to old, and the sort of mechanisms that underpin that. Is that how you define aging? Or if someone just says plainly to you, Andrew, what is aging? How do you go about sort of describing that, unpacking that?
I mean, ultimately, Matt is exactly right. Aging is a biological process. But I think you can take a step back. And actually, the way that I usually define it at first is exactly that definition that I've just given you implicitly in that graph. The rate of aging as a human being is effectively the change in your risk of death with time. So that risk of death doubling every 8 years, that's a quantity known as the mortality rate doubling time. So just to unpack that, mortality rate is obviously risk of death, and doubling time is just how long it takes for that risk of death to double. And for human beings, that's 8 years. And what I think is really remarkable about that number, obviously, life expectancy has changed dramatically. It's roughly doubled in the last 2 centuries. There's huge disparities in life expectancy around the world between the rich and the poor in countries. There's all sorts to unpack in the sort of, the understanding, the statistics of life expectancy. But if you plucked someone, from the depths of history, some undiscovered tribe from the Amazon rainforest, someone from any country around the modern world, what you'd find is that although their absolute risk of death might change quite substantially between those populations, that rate of doubling of risk of death is basically a constant. It's a fact of human biology. And I think that's a really good place to start, a much simpler place to understand the biology of aging from than thinking about the nitty-gritty of what's actually happening in our cells. And this isn't a universal fact of sort of biology across species. So there are some animals, obviously, that live a lot shorter than us. You might think of like a mouse or something like that. That's got a mortality rate doubling time of about a few months. And that's why, mice only typically live 2-3 years on average. But then there are other animals that have much longer mortality rate doubling times than us. And in fact, there are some animals that don't really have a mortality rate doubling time. What does that mean? Well, for example, if you look at an animal like a giant tortoise, that has a risk of death that stays essentially constant with age once it becomes an adult. And that means that, it's got a sort of 1 or 2% chance of dying every year. It's running that same risk of death every single year. And so it doesn't get this exponential increase in mortality. And that means in this statistical sense, and actually I think in a biological sense as well, those tortoises don't age.
Yeah, I want to come to longevity escape velocity at some point. Perhaps you can remind me later on if I forget to come back to that. But while we're on this, what is it about aging that you care so much about? I think I've heard you describe it as the largest humanitarian challenge out there. And I'm sure that there's probably someone thinking, isn't aging just part of being a human? You're born, you come into the world, you age, you die. Should we really be scared of death and focusing all of our attention on just trying to live longer? How would you sort of go about unpacking that?
Yeah, it's a big one. And I think that's something I truly believe. I talk about it in the book as well, this idea of our largest humanitarian challenge. And I think it does sound slightly strange the first time you come across it. It's difficult for me because I've been thinking about this stuff for, a decade or more. I've sort of indoctrinated myself, I guess. But to go back to the very beginning, let's think about, what is it that the aging process causes? Well, it causes diseases. It causes things like cancer and heart disease and stroke. It causes things like dementia. So these are things that have a huge impact on quality of life. You don't just, get cancer and then, wake up one morning or not wake up one morning and you're dead. And it's the same with a heart attack. I think a lot of people have this sort of image that you suddenly get a heart attack, you fall over clutching your chest and then you're just dead. But what often happens is that these diseases are preceded by years, maybe even decades of suffering specific to that disease. So your heart is slowly failing, you're losing your independence, you're less able to get on with your life. If you've got something like cancer, the treatment can be quite grueling. Chemotherapy is no fun, as anyone who's ever had it will tell you. And so as a result, there's a huge amount of suffering bundled up with these causes of death. Then there's the stuff that we don't necessarily call diseases. So something like frailty. You might go to your doctor, you've got significantly reduced muscle mass in old age, you're much less able to get up the stairs, or, you can't open jars. These things that seem like mild inconveniences can really affect your quality of life as they sort of spread out and affect more and more of your everyday activities. But there's not really a strict definition. If you lose a huge amount of muscle mass, you might be diagnosed with a condition called sarcopenia. But by and large, people who lose muscle mass as they age, that's just considered a natural part of the aging process. And there's this huge collection of things, cognitive decline that isn't quite bad enough to be dementia, a little bit of hearing loss, all of these things that slowly again erode away your quality of life, but might not necessarily have a medical diagnosis. And, unfortunately, all of these things are quite difficult to statistically categorize because we don't have a big, neat list of all the different, things that happen to people as they get older. So I do end up gravitating back towards death because death is a nice, easy, what's called an endpoint in medical trials. You're either dead or you're not. There's no ambiguity about that. So what we can do is we can tot up all of the different ways that people can die. And we can look at this mortality rate doubling time. And we can do some statistics. And if you do that, do those statistics, you find that over 2/3 of deaths around the world. So of the 150,000 people who die every single day in all the countries of the world, not just the rich ones, more than 100,000 people every single day die because of aging. Now, you might think, I'm not worried about dying. I'm not going to be around after all. So, what is there to worry about? Fair enough. That's a philosophical discussion. Perhaps we can park for a minute. But the fact is that, as I've explained, all of these deaths are preceded by these years of degeneration, of frailty, of suffering specific to the diseases you're suffering from. And that's why I think that aging is the world's greatest humanitarian challenge. And of course, this over 2/3 number is getting bigger and bigger and bigger. If we concentrate on the rich countries where people are living longer and longer, and in many countries, over 90% of deaths are caused by aging. And so when you get to that sort of level, it is our single biggest cause of death. I would argue our single biggest cause of suffering. And that's really what motivates me to want to do something about it.
Let's double click on that word cause. So I think a lot of people will be familiar with cardiovascular disease or various types of cancers. And I guess sort of up until this point, definitely in the kind of public conversation anyway, most of the conversation has been on things like, optimize your blood pressure or your cholesterol, these sort of known or established risk factors. And we think about those things as sort of causing these diseases. But you're looking at this through a sort of different lens, the lens of the biology of aging. So can you kind of speak to how aging is actually the root cause that underpins these many diseases that we see and that are affecting people and causing that suffering that you just spoke to?
Yeah, and I think the way that doctors and scientists often talk about this is they talk about something called a risk factor. So these are things that increase your risk of a given disease. And of course, risk factors can take a whole variety of forms. You mentioned blood pressure, cholesterol. These things can all be risk factors. There's also the lifestyle stuff. So smoking is a risk factor. A bad diet is a risk factor, and so on and so on. But the single greatest risk factor for most diseases is simply being old. And to give a very sort of simple illustrative example of this, imagine heart disease. You can be a chain smoking, heavy drinking, overweight, terrible eating, never exercising 30-something, and you'll almost certainly not have a heart attack. Whereas you can be the cleanest living, healthiest 80-year-old who's had an absolutely perfect life from a sort of health advice point of view, and you can just get a heart attack out of the blue. And there are a variety of different reasons for this. Some of them are biological. They're changes that happen in our biology with age. Some of them are simply the accumulation of time. Like if you think about atherosclerosis, the accumulation of these fatty plaques in our arteries, it just takes, decades for those plaques to slowly accumulate. There have been these autopsy studies done on people like teenagers who die in car accidents. And if you look inside their arteries and veins, they already have some of these plaques. They're just very small, far too small to ever think about becoming a problem. But over the years and decades, slowly, some combination of accumulated time, so these things sort of build up, and things like increases in inflammation, which I'm sure we'll talk about, go on to make these things much, much larger problems.
What do you think is the kind of maximum human lifespan? What's the potential here? So we're going to talk about a lot of different innovations, and we'll no doubt touch on lifestyle, but with all of the exciting stuff that's happening and your understanding of what's causing aging and the field's understanding of that, what do you think is the limit? Is there a ceiling? Are we talking about getting more people to live to, say, 120 years, which I guess around there many may say is the kind of maximum human lifespan? Or do you think that may not be the ceiling and we could push up to 150, 200 years? And also, how long do you think it will take to kind of get to those numbers if you think they're possible?
I think we're going to accidentally end up talking about longevity escape velocity in this question, actually, because I think that it's sort of it's an ill-defined question, which I guess is a scientist's polite way of saying it's not necessarily a very easy question to answer. The trouble is that I think, if you were to give someone the best, absolute best dietary advice, health, healthcare, all of these different things, probably, given that we've had so many billions of people alive on Earth, 120, maybe 130 is probably the maximum life expectancy we could look at. Because, throughout all of human history, in terms of, people who've got well recorded, they've got a birth certificate, they've got a death certificate from a sufficiently modern time that we can be fairly sure it was true. 122 and a bit seems to be the maximum human lifespan. And in fact, no one's even approached that. That record, astonishingly, was set in 1997. And in the intervening sort of 2.5 decades, we still haven't got anywhere near. And in fact, the current oldest person is going to have to live quite a few more years. It's going to be quite unlikely that they're going to beat this record set by Jean Calment. So I think even with the absolute best current understanding and current interventions that we have available to us, that probably is something of a ceiling. However, I think that that's a bit of a sort of misnomer, a bit of something that we shouldn't be seeing as a ceiling from the perspective of people who are alive today. And the reason for that is that we just don't know what the developments are going to be in aging biology. And these are a whole new paradigm in medicine. So what we do at the moment, most of medicine is treating these endpoints. It's treating cancer, it's treating heart disease. Hopefully sometimes it's preventing cancer or preventing heart disease, but ultimately we're often targeting these individual diseases. And if you get someone who's 80 and you cure their cancer because of, amazing modern cancer medicines, not to detract from, how incredible that achievement is, they're still going to be in an aged body. They're going to be, in fact, in an aged body that's been battered by 5 years of cancer treatment, maybe to get rid of that cancer. So they've probably got a bit of heart disease going on. They're probably, edging into the first part of dementia and so on and so on. So there's something else waiting in the wings to kill them just a few years later. Whereas if we can treat the aging process, if we can understand what gives rise to all of these diseases in the first place, we can potentially defer their risk of death. We can push these diseases further back into the future. And I think ultimately, the way that you've got to think about this is that looking around the animal kingdom, I've already mentioned tortoises. There's also fish. There are some lizards. There are quite a few animals that have this property where their risk of death doesn't change with time. And this is a property that's known as negligible senescence. Negligible, not very much. Senescence is just the biological term for aging. And what this shows us is there's no biological reason why we have to grow old. There's no biological reason we have to become more frail. Although I've talked about risk of death, again, I focus on that because it's nice and easy to do the statistics on. These tortoises, they stay active until late in life. They stay, able to reproduce. All of their functions remain until the very last few years of their life. They just seem to carry on and suddenly drop off a cliff. And so, the first thing to say is that there's all this aging medicine. We've talked a lot about lifespan. It's about increasing health span. It's about increasing the amount of time we're able to do what we want. It's about increasing the amount of our lives which we are free from disease. But ultimately, if you're not getting those diseases, and these diseases are causing the majority of death in particularly the rich countries, but actually all around the world, if you're not getting the cancer, you're not getting the dementia, you are going to die a bit later, almost as a side effect of improving how healthy you are. Now, to sort of rewind back, I mentioned longevity escape velocity right at the start of this answer. The reason that becomes relevant is imagine the average person in a rich country these days is about 40 years old. So imagine you're that average person. I am almost that average person, actually. I've got, fingers crossed, I've got something like 40 or 50 years of life left ahead of me, if nothing in particularly special happens to life expectancy. And again, if we follow all the good health advice, if we get a bit lucky, we can probably hope to live into our 90s. What does that mean? Well, that means there's 50 years of biology development ahead of us. And we're already in a position, I'm sure we'll talk about some of this, as we go through the conversation. We have a few longevity drugs that could be available in the next 5 or 10 years. I think that's, fairly likely that something is going to make it in the next 5 or 10 years. And that means because we're going to live long enough to benefit from that first generation of longevity drugs, it might only add a few years to life expectancy. It might just keep us healthy a little bit longer. But that means we've got a little bit more time on top of those 50 years for biologists to carry on doing that development. And the idea of longevity escape velocity is this concept that if you're living in a period where life expectancy is increasing at 1 year per year, so you can expect to live 1 year longer per year that you live, then ultimately you can sort of think about it as your funeral rushing off over the horizon at the same speed that you're chasing it. And so ultimately you'll never get there. Now, of course, this is a hypothetical idea. No one's saying it's necessarily going to happen, in the next 5 or 10 or 20 or even 50 years. But this idea that by buying yourself more time, you can extend your lifespan by allowing yourself to benefit from more scientific developments, that's a really powerful idea. And whether that means we're all going to live to 1,000 or, whatever crazy number some people might pluck out of the air, it certainly means it's very hard to put a number on how long people in roughly middle age, in the modern world can expect to live. Because it really depends crucially on how lucky we get with a lot of this aging biology development, and crucially how much funding we put into this research. Because ultimately it's, staying alive long enough, staying healthy, following all the health advice in order to live long enough to benefit from these treatments to then live longer, and so on and so on and so on. And who knows where we might end up.
Yeah, I really liked a quote from your book that said, at the moment, every year you're alive sees your probable date of death recede a few months into the future.
Yeah, it's a crazy thing to think about, isn't it? That, you know, it's literally just by effectively doing nothing, watching medicine improve in the background. That's what's going on.
Those other animals that have very long lifespans, and it seems like they have a very short period of disease. They have very good health spans. How much of that could be down to the genetic differences between humans and those animals? And I guess where I'm getting with this question is, how much of our overall, I guess, lifespan is dictated by the genes that we have versus things that we could control through the various behaviors that you mentioned, like not smoking and eating well, and then potentially some of these other therapeutics and innovations that become available over time?
Yeah, well, the answer for the animals question is actually very easy. 100% of the difference between the lifespan between these animals is determined genetically. Like, fundamentally, the reason a mouse lives a few years, the reason that we live 70 years or 80 years or 90 years, the reason that something like a bowhead whale can live a couple of 100 years is written in our genes, because we all have ultimately very much the same machinery going on inside our cells. They're just translating those genetic differences into lifespan differences. And of course, the reason these animals have evolved very different lifespans is just because they're in very different evolutionary environments. And so the optimal lifespan to propagate their genes to the next generation is very different in those situations. So that's 100% genetic. When you're thinking about what's going on within species, though, actually, the results are quite interesting. And in humans, there's a little bit of controversy because it depends a bit on how you do the maths. But ultimately, something like at the low end, if you do the maths in one way, you can get 5% of your lifespan is determined genetically. And at the higher end, if you do the maths in a slightly different way, it's something like 20-25%. And what that means is that somewhere between 75 and 95% of how long you live is determined by two things. One of them's lifestyle, and the other one is just luck. And you've got to think a little bit about that luck aspect as well. You know, obviously, it means there's a lot we can do. It's a very empowering thought, first of all, because it means, you know, by eating well, by exercising, by doing all these things that we know are supposed to be good for us, then you can have a huge effect on how long you're going to live. But it also means there is a large degree of luck, because, you know, whether or not a particular cell happens to accumulate the wrong combination of mutations and become a cancer, you know, there's a huge degree of just random chance in those kinds of processes. The only place where this sort of differs, this sort of broad population level trend, is in people who live a very, very long time. So let's imagine your parents, say they made it to between sort of 60 and 90 years old. Ultimately, you don't have to look at their lifespan and see that as any kind of ceiling or any kind of determinant of how long you're going to live. Because, as I said, somewhere between 75 and 95% of it is either within your control from lifestyle or just luck. But then if you look at the people who live incredibly long lives, people who make it into their 90s, their hundreds, or even their 110s, these centenarians and super centenarians, as they're called, that does seem to run in families to a much greater extent. So if you've got a parent or a sibling who made it to 100, your chances of doing it yourself are 10 times greater than people in the general population. So it seems that people who make it into these incredibly old ages do have some kind of protective genetics that we're beginning to unravel. It's not entirely clear what that might be. But for most of us, the genetic contribution is surprisingly small.
Yeah, I was interested in, I think it was the latter half of your book. You did write about potentially being able to use some of those learnings or understandings of protective genes in the future through different therapies. Was I understanding correctly? We could add or sort of edit genes based on our understanding of these kind of centenarians, long-living people, and use that as another kind of innovative therapy to extend life.
Yeah, and I think actually gene editing, it sounds really sci-fi, doesn't it, that we can go in and edit our DNA? But actually, I think this is something that's nearer term than a lot of people think. One reason it's nearer term is just that if you live long enough, as we were saying earlier, to benefit from the early sort of normal, quote-unquote, drug-based therapy, something like a senolytic drug, which I'm sure we'll talk about, that can then make you live a little bit longer, then you can start to benefit from these things that sound like they're further in the future. But actually, you know, when you look at gene therapy, there are already gene therapies in the clinic. We're living in this fascinating, exciting time. At the moment, the way the gene therapies work is they're normally given to people with very serious genetic conditions. So they're people who are born with a particular, effectively genetic problem that then we can go in and fix that problem and massively extend their lifespan. And the reason that we're willing to do that is, these therapies, they are experimental. We're only just learning how to edit genes within live human beings rather than cells in a dish or animals in the lab. And so we're taking baby steps, and it's going to be people with really serious conditions who are sort of willing to take a punt on these experimental therapies effectively. But I think a really good example of a gene that is overrepresented in centenarians and something that we could think about editing in the relatively near term is the ApoE gene, which I'm sure a lot of your listeners have heard of. This is a gene that has 3 different variants that are commonly around in the population. ApoE3 is the normal, sort of quote unquote normal one. It's the most prevalent. There's also ApoE2 and ApoE4. And we know that people who've got a copy of ApoE4, or worst of all, 2 copies of ApoE4, so one from their mum, one from their dad, are at significantly increased risk of a variety of age-related diseases. But what's really fascinating about these 3 variants of the ApoE gene is that actually they differ by a single genetic letter. So our DNA is obviously made up of these bases. They're A's, T's, C's, and G's is how we refer to them. And it's just a single letter difference at a particular point in that gene is the difference between ApoE3 and ApoE4 and ApoE3 and ApoE2. They're in slightly different places. But nonetheless, this is quite an easy gene edit. So if people have heard of CRISPR, this is this technology that can go in and cut your DNA in any chosen sort of place. You guide this enzyme in that's going to snip the DNA with a little sequence, and so you can target it very specifically within the DNA. It's a very exciting technology. There's a modified version of CRISPR, which is called base editing, where again you give it this target, so it goes in and finds a particular stretch of DNA, but rather than just slicing it, it can switch one DNA letter for another. And actually, the paper in which this technology was introduced a few years ago, as an example, they got some cells in a dish and they edited their ApoE genes to demonstrate that was a possibility. And so you can well imagine this is a relatively low risk gene therapy, because it's the sort of thing where you don't have to edit every single cell in the body. That's a common problem with gene therapy, because there are 37 trillion cells in your body. It's very, very hard to get every last one of them. But if you don't need to edit every single cell, it's something that might be worth a punt. And we also know that people have, as we just were discussing, you can have 2 copies of ApoE4. You can have ApoE3 and ApoE4. That's a bit better. So say you are someone with 2 copies of ApoE4 and I just got rid of a small amount of it, that would probably slightly improve your prognosis. And so we know that it's possible for these genes to coexist. So it really does look like quite a tempting target to just try this out and see if we can improve people's life expectancy. And of course, we can carry on mining the genomes of centenarians and supercentenarians to try and work out what they're doing that the rest of us aren't. And hopefully, it's not going to be decades and decades before we can start thinking about doing some of these things as gene therapies.
Yeah, I think Chris Hemsworth brought a lot of awareness to ApoE4 and how that can increase risk of Alzheimer's dementia. How far off is that type of therapy for ApoE4? Is that something that has been tested in humans?
It hasn't been tested in humans yet, as far as I'm aware, but I think base editing is being tested for, as I said, these sort of serious genetic diseases. And I think that's really, it's an important pathway to sort of follow for all of these aging-related treatments. So a lot of these things, if you come up with a drug that can treat aging, it can often treat a specific age-related disease, or sometimes even a not particularly age-related disease with the same technology. And what you often find in medicine is that these things, they sort of trickle down through the levels of disease severity. So you start off giving them to people who've got a really serious disease, who are sort of willing to try something out because they haven't really got any treatment alternatives. But as we get more and more confident doing these gene therapies, we make sure there aren't any what are called off-target effects, so places where it edits the wrong bit of the genome. There aren't any increased risk of cancer in the people who are getting these genetic edits and so on. Then you can start spreading the net more widely. So it's a bit hard to give an exact timescale in years, but I really don't feel like it's that far away, in the sense that, as I say, there are already some gene therapies in the clinic. I think there's some base editing stuff currently in clinical trials. You could well imagine that in 5 or 10 or 15 years, easily soon enough to benefit most people alive today, we could be thinking about doing this stuff. And I think it's just really important that we sort of realize collectively that this is a possibility and sort of have this conversation ahead of time, because this gene editing does sound super futuristic, but it's coming sooner than I think a lot of people realize.
Yeah, I just had a series on lipids with Dr. Thomas Dayspring, and one thing that we focused on a little bit was different gene mutations that can affect the level of these ApoB containing lipoproteins in circulation, LDLs being the predominant ApoB containing lipoprotein. And he spoke about people who have a PCSK9 gain of function. And so if they have that sort of genetic variant that results in sort of less of the LDL receptors, and then they end up with a much higher ApoB level in circulation and have dramatically increased risk of atherosclerosis. And if untreated, depending on sort of the level of that, some of them are experiencing cardiovascular events, teenage, early 20s, so like very early in life. And I did read both in your book and I'd read some sort of press releases that there were groups looking at least in primates, maybe at this stage, I'm not sure if it's moved to humans, but looking at gene editing using CRISPR, potentially in that sort of context.
Yeah, I think this is super exciting. So the way this research seems to have gone, I came at the sort of discovery of PCSK9 from the other direction in the book. So this discovery that there was this population, mainly of African Americans, who seem to have a deficiency of PCSK9, and looking at them, there's nothing wrong with them. They're living perfectly good life expectancies. In fact, slightly improved life expectancies, perhaps, because they're not getting a lot of these cardiovascular problems. They're at much lower risk of heart disease. They're at much lower risk of stroke, much lower risk of some kinds of dementia. This is, really quite an incredible thing for a single gene mutation. And so the idea is that we can go in and inhibit PCSK9 and massively reduce the levels of cholesterol in the blood. And obviously, this is also another fascinating example. There's this big debate about how low should we take cholesterol levels. Well, these are walking human examples of people with super duper low cholesterol, and they seem fine. Like, it's not to say there's not any sort of subtle issues that we haven't picked up on, because it's obviously very difficult to compare between populations, but they do seem to be all right. So that's sort of a compelling reason to think that lowering LDL, lowering ApoB is a really good thing. So the way this started out is the classic way that medicine's been working for, a few decades now. This gene was identified and scientists found drugs that can inhibit that gene. They found antibodies that can go in and deal with that PCSK9. But the next sort of generation of this therapy, and I think this has just been approved, is something called RNA interference technology, a silencing RNA. So what can happen is let's rewind a bit to sort of high school biology here. When a gene is expressed, your DNA is full of these sections, the genes. These are the things that manufacture the proteins that actually do all the work in the cell. And so PCSK9 is a protein. The PCSK9 gene starts out as DNA. And if you want to make more of that protein, that DNA gets copied into a format called RNA, very similar molecule, as you can tell from the very similar name. Then RNA goes on to make the protein. And these silencing RNAs, they go in and they basically degrade that RNA intermediate. And so the protein never gets made. So that massively reduces the level of PCSK9. Now, this is much, much better than a drug or an antibody, because it seems to last, I think, 9 months even. So patients can go in for an injection and then not have to bother, thinking about taking their tablets every morning, remembering to take their statins or whatever it might be. So that's a great thing in and of itself. But the next stage, and there is already, as you say, a company, I think, doing trials in primates who are looking to disable this gene at the DNA level. So we're not going to interfere in the RNA. We're going to go in, we're going to switch off that gene just permanently. And that's a hugely exciting idea, because if we could then just lower cholesterol levels without you having to, necessarily think so much about it, certainly without having to remembering to take a pill, not even having to go into your doctor every 6 months and get an injection, that would be so cool. And I think, as I sort of alluded earlier, this is really exciting because we've got a human population where we can see this gene doesn't appear to do anything terrible in the long term. That's something I'd be personally quite tempted to get, to be honest.
What are the kind of potential problems if someone's thinking, messing around with genes, it sounds like that could be reasonably risky. What would the, I guess, likely adverse or side effects be, potentially from editing genes? And is it something that once you've done, is it reversible?
I think that's a really important question, is the reversibility aspect, because if you've done something like a base edit, and so you've got a particular stretch of DNA, you go in and change a particular letter, there's no reason in principle you can't go and change that letter back. If you were to do some more complicated edit, perhaps that might be more difficult, and that's definitely something to think about. But I think in terms of what the side effects might be, unfortunately, the ultimate answer is how long is a piece of string. It's going to depend very much on the specific gene, what that gene does. And what you find in biology is that you see a lot of headlines, and this persists to this day, of, we found the gene for X, and X is often, some incredibly complicated and specific trait. But obviously, in reality, genes are much, much more complicated than that. Genes can have, it's called pleiotropy. And that pleiotropy just means that they can have a variety of different functions perhaps in different cells around the body, perhaps in different times during your development, during your lifespan. They can do lots and lots of different things. And so when you disable a particular gene, you might find that, you've done a great thing for the health of your kidneys, but a terrible thing for the health of your lungs, or just various trade-offs like this. I think the only way to answer these questions really is to try these things out, because ultimately ultimately biology is just so complicated, it's hard to make a priori predictions. And there are certainly some things where it would be, a good idea to make sure that the thing is reversible. It's not the case that once you've made the edit, it's somehow irreversible, because obviously if you can go in and change DNA, you can go in and change it back. But I think, and the other thing to say about this actually is that, editing DNA it can sound a bit sinister. It can sound a bit sort of futuristic, high tech. It's got all kinds of connotations, but actually drugs have all kinds of what are called off-target effects as well. So where they target other parts of the body, or they interact with another molecule they're not supposed to interact with and cause all kinds of side effects. In a way, genes are much less likely to have these side effects, because if you're aiming a drug at a particular gene, it might hit a bunch of other genes. But if you aim a gene therapy at a particular gene, well, it's guaranteed to only hit that particular gene, right? Assuming, we can do the edit accurately and so on. So there's definitely potential for these things to be more precise than drugs rather than less. But unfortunately, as I say, it's very much going to be on a case-by-case basis.
Yeah, it's exciting for people to kind of be aware of and kind of watch this space, I guess. I want to ask you about senolytics and also cellular reprogramming. But before we get to those sort of areas of innovation in this field, I've had a couple of previous episodes where drugs like rapamycin and metformin have come up. And they're probably, I guess, the two compounds that get the most airtime, I guess, in this space, probably due to the sort of trials that are underway or hopefully underway soon. What are your views on these two compounds and their potential utility within the kind of pursuit of slowing down aging?
I think they're both really exciting, but unfortunately my boring answer is going to be that we're going to have to wait for the results of the trials to find out. Metformin is in this really frustrating place where we've got, there's enough conflicting results that you can make any case that you want in any particular direction. I think the most exciting result that we've got in favor of metformin being a longevity drug is this trial that I think you actually spoke about with Matt Caberline, which is this one where they had a bunch of, it was Scottish people actually, so it was a UK medical records study, and they looked at people taking metformin, they looked at people taking another class of diabetes drug called sulfonylureas, and ultimately the purpose of the trial was to compare these two diabetes drugs, but also to be really, really sort of belt and braces statistically certain. They included this control group who had people not taking metformin. And of course, the reason they weren't taking metformin is because they weren't diabetic. And you would expect in general that diabetics would live less long than non-diabetics. And the reason you'd think that is often they have more heart problems. They have all sorts of complications that come along with the diabetes. They're often less fit. They're often more overweight. That might be contributing to their disease and so on. So you'd expect that those diabetics would live less long than the non-diabetics. But by giving them metformin, their lifespan actually seemed to be increased by a small amount. It was an increase of, I think, something like a few months. So not earth-shattering. This isn't suddenly going to make us all immortal. But at the same time, given that you're looking at a population who you'd naturally expect to have a lower life expectancy, it's quite an interesting result. But the challenge with this, and there have been other studies that have come up with slightly different answers. There are studies in mice and all kinds of things that you can point to for understanding the biochemical pathways of metformin is a nightmare because it is the filthiest drug around. It hits loads and loads of different parts of the body. It even affects the microbiome. So there are just so many ways in which this could be affecting our biology. But I think that the thing that always undermines these observational studies, as they're called, where rather than giving people a drug at random, you just look and see what happened historically. There might be other reasons that people who are taking metformin, diabetics taking metformin, might live longer. So for example, if you're diabetic, you're more likely to visit your doctor more often and get various tests. So if you've got high blood pressure, your doctor's more likely to spot it. If you've got anything else, sort of a nascent issue, again, your doctor's more likely to spot it. So it could be that those diabetics are on metformin, they're getting more attention from the healthcare system than all the other people who, think they're healthy, but actually might have some underlying issue. And that's why those people are dying sooner. And that's ultimately why we really need the TAME trial. And that's this trial where they're actually going to do a proper randomized trial where they give half the people metformin, half the people a placebo pill that doesn't have any effect, and see who lives longer, who gets diseases later, and so on, and see if metformin really does have that effect. And I think, unfortunately, the data for metformin are just so confusing that ultimately we're going to have to wait. That trial really needs to be funded. It's super important. Rapamycin, I think, is probably the more exciting drug in the longer term. I think the reason that when they were trying to decide what drug to use for the TAME trial, you've got to bear in mind this is the first ever trial of a drug that's aimed at the biology of aging specifically, rather than aimed at a particular disease. And so when they were trying to choose the drug, they wanted to choose something that had a good chance of working. Otherwise, what's the point? But you also want to choose something that's quite safe. And the real challenge with rapamycin is it's got a whole sort of suite of side effects. Actually, I think this is sometimes a bit overblown. And to give an example of this, during the pandemic, I know a scientist who was trying to get rapamycin trialed in care homes. because they were hoping that people who are given rapamycin would have, that they'd effectively have the aging of their immune system reduced a little bit, and so it might reduce mortality from COVID-19. But the challenge was, when she was approaching, doctors and care homes and the people trying to organise the trial, if doctors know one thing about rapamycin, they know it's an immune suppressant, because how have they prescribed it? If they have prescribed it in their careers, it's to people who've had transplants in order to suppress their immune system in order to try and stop the patient rejecting that transplant. And so, you're going around to a bunch of care homes with your pot of rapamycin and saying, I want to give all these old people this immune suppressing drug that the only time you've ever used it in your career is to suppress the immune system during a time when the deadliest pandemic in the last 100 years is going around. Obviously, there's a degree of skepticism. I think the crucial thing with a lot of the rapamycin side effects, that's the immune suppression, that's the mouth ulcers, that all these various things that are observed in trials, the doses that are given to suppress the immune system of transplant patients are much higher than the doses that are thought to be perhaps beneficial from a longevity point of view. Because you don't want to be suppressing the immune system. You want to be just sort of tamping down the mTOR, the target that the rapamycin hits, a little bit, and that seems to rejuvenate the immune system. Paradoxically, a small amount of an immune suppressant seems to make the immune system stronger in the trials that we've seen. But I can see why, if you're playing the TAME trial, you're playing the first ever sort of showcase of aging biology. You don't want to choose something that might be giving a bunch of people, really weird side effects. So you don't want to choose something that the medical community are immediately going to be very skeptical of. So I hope that as we understand more about rapamycin, that as well, have its own effectively TAME trial. But I can see why they decided to go for metformin first, just because rapamycin is it's a bit of a spicier drug, shall we say.
Yeah, there seems to be quite a community of Rapa devotees online who are sort of, I guess, hedging their bets. They're getting rapamycin off label, and there's all these different dosing protocols that you can come across online, and extrapolating from different animal studies and studies looking at, I believe, patients with cancer. How do you feel about that? Like, if someone's listening right now and they think, well, I can justify the cost of this. I appreciate what you're saying, Andrew, but I don't really want to wait 5, 10, 15 years and then find out that this has a anti-aging effect and I could have started much earlier. How would you sort of encourage someone to think about this?
I think it's a really hard question, actually. It's something that I've sort of thought about myself, and I talk a bit about at the end of the book, actually. This idea that in my ideal world, I'm not the right sort of doctor to be giving medical advice, as you said at the start of this talk, this interview. I'm a physics PhD, so none of this is medical advice, just to preface all that. But I'm really frustrated that we haven't got a mechanism through which people who are doctors, people who are experts in aging, a variety of people can come together and make some kind of recommendation based on the sort of costs and benefits that we know to exist. Because we've already mentioned I'm 37. I am sufficiently young that I'm happy to wait 10 or 15 years. If my risk of death this year is 1 in 1,000, I'm happy just to let that slide. The other really cool thing about rapamycin is that the trial in which it really sort of came to prominence, it was started in mice at very, very late ages. I think you and Matt talked about this as well. The trial was started, and actually it's a really fascinating story. It's because of effectively a problem with the trial protocol. They were giving these mice rapamycin in their water, and they did a blood test, and they discovered there wasn't any rapamycin in their blood. So they realized the way that they were feeding the mice the rapamycin wasn't biocompatible, wasn't bioavailable. And so they spent about 2 years trying to work out a way to reformulate the rapamycin to get it into the mice. And when they got to that point in the experiment, I think a lot of people were just like, well, these mice are old. Like, what is the point? If you've got a drug that slows down aging and you start it really late in life, you can imagine just slowing down that last little bit of life isn't going to make a huge deal of difference in terms of health or lifespan. But the astonishing result was that these effectively sort of 60-year-old, in human terms, mice were living longer, they were living healthier, they had less disease, and we now know there are this huge sort of suite of functional benefits from taking rapamycin. So if you are, if you're not even a particularly younger person, even if you're 50 or something, you can afford to wait the 5, 10, 15 years, because it does appear that you could take rapamycin starting at 60, 65, 70 and probably still see a lot of the benefits. So there's no need to sort of jump the gun in that respect. Now, the reason I want to get this sort of collection of eggheads together and come up with some kind of recommendations is because if I was 65 or 70, then there's a much more interesting, sort of calculation to be made, because by the time you're 65, your risk of death in that year is about 1%. That's still not huge. You'd still expect to live quite a few more years if that were to continue, but it's enough that you're starting to think, well, rapamycin at low doses, we know it's fairly safe. We've got this sort of semi-anecdotal evidence from people trying it out. And what's really frustrating is there is this community of people who want to try it out. I think there's probably an even larger community of people who, if they were sort of made aware that this was a thing, and were also made aware that they could take part in a trial that might improve their life expectancy, might improve their health span, but also would provide this knowledge for future generations, because this is a super important thing. It could be hugely beneficial. If it turns out rapamycin slows down aging, it's an off-patent drug, it's super cheap. Like, we could just roll this stuff out almost immediately. So there's this huge benefit to doing a trial. And it's really annoying to me that there isn't some mechanism by which we can pull together all the sort of biohackers and the keen older people who'd like to understand and contribute to this research and give them a protocol that's as evidence-based as we can, given the situation. Perhaps we could even give a variety of protocols if enough people were willing to take part in the trial. And then we could actually find out for real whether this thing works or not, while giving people the opportunity to do that experiment. Because the real problem with a lot of this sort of self-experimentation is it's very hard to know. Often people order these drugs online, so you're not quite sure, are you actually getting rapamycin? Has it been adulterated with anything? Is it pure, et cetera, et cetera? What's the dose? Have I got the right quantity of this stuff according to the protocol that I'm following? And it's all done in this quite an erratic way. Whereas if it were done within a slightly more formalized setting, not only would it be safer for the patients, it would also allow us to collect data from that trial and understand what's really going on. But it's a really difficult thing. I don't know what the answer is if you're an older person listening to this, thinking about taking a drug like rapamycin or metformin, because it's not super clear cut. They're not obviously super dangerous drugs, and your risk of death is already starting to climb a bit. And I really wish, some collection of people who, have more than just a PhD in physics to rub between them could, come up with some kind of recommendation for this, because it's, I actually think there's a sort of wider problem here as well, which is that as we continue to accumulate evidence on aging biology, there's going to be a whole swathe of interventions. You've already mentioned, senolytics, reprogramming. There are already loads of supplements and, these sort of flavanols and polyphenols and spermidine and all these things that have some degree of evidence for them. Not nothing, but equally not the perfect randomized controlled trial that a nerd like me might like to see before they start taking it. And so there needs to be some sort of framework in which we can say, okay, on balance, this is sufficiently safe and might have enough evidence of benefit that we can think about, giving people the evidence to make the decision for themselves. Not necessarily to the point of recommending it, not necessarily to the point of doctors handing it out, but giving people the option. Because I think that, doing nothing is also a choice medically. And I think that isn't explicit enough in the way that we currently deal with medicine. And that's because most of it, as I already mentioned, is about, you come in, you've got a lump, we do a biopsy, it turns out it's cancer, we treat the cancer. It's very reactive medicine. Whereas if you're trying to do proactive, preventative medicine, there's always a degree of uncertainty until you've done that, perfect trial where you started everyone at the age of 20 on this drug and waited until they got to the age of 100. We'll definitely know the answer in the year 2200, but that might be too late for everybody who's currently alive listening to this. And so we really need to come up with a way of assessing these costs and benefits and making some kind of sense of the uncertainty. And I don't think that's something that we've grappled with as a medical community, as a scientific community, or as a society, because it's just a whole new paradigm with the way we do medicine.
Yeah, I think some sort of independent committee would be extremely helpful. And it also makes me think about conflicts of interest, I guess, in this space. And there seems to be a lot of blurring between science and private corporations, and there's all these startups popping up overnight. And something that I've been thinking a lot about is how do we ensure that the conflicts of interest are not leading to exaggerated claims about, novel products and or services that slow or reverse aging?
Yeah, it's very, very difficult. And I think this is actually a case where it would be good to have some kind of centralized body. There's obviously the FDA, but you get, by the time something's ready to be FDA approved, it's really, jumped through an awful lot of hoops, and they're very, very cautious, which is, perhaps a good thing. You can debate exactly how cautious and how experimental the FDA should be. That's a whole nother podcast. But there does need to be some kind of independent way of assessing these things. And there's all kinds of claims going on. There are loads of different companies who'll measure your biological age, whatever that might mean, depending on the test they're offering. There are people who offer like customized supplements. There are so many different things on the market. And then the sort of supplement market is an entirely different beast when it comes to regulation, because often it's effectively unregulated. As long as your supplement doesn't kill people acutely, it doesn't kill them in the short term, it's that you're able to hand these things out with a regulation that's far more like a food product than it is like a medicine. And so, yeah, I just think we're in a difficult period at the moment, because again, it's this whole new paradigm of medicine. It's just not something that we're used to dealing with.
Yeah, and I think the companies realize that the demand for these products is there. And I'm sure you saw the blueprint that Bryan Johnson, I believe, put out. And, I found that quite interesting, both what he was doing and, I was interested with regards to the evidence that maybe supports all of it, and whether there even could be interactions between some of the things that he's taking and the effect that could potentially have on health. But I think what interests me most was the amount of social sharing, discussion, conversation. People were really, really interested in the things that this guy was doing, which, speaks to the large demand that exists. And I just think that companies are aware of that and are going to capitalize on that, irrespective of whether the science is here today.
Yeah, I think that's right. I actually made a YouTube video about Bryan Johnson's blueprint months and months ago, because I saw a Twitter thread on it go viral. And that video has just kept on performing, and all the hits are coming through search. So people are clearly looking for Bryan Johnson blueprint on YouTube and coming across my video. And there's this, it's obviously very hard to tell because the comments aren't always a representative sample of your population of viewers. But there are some really avid Bryan Johnson defenders, because my video is quite like, I would say, measured and cautious and skeptical, but perhaps, others may disagree. But they're really avidly, I think a lot of people want this stuff to be true, because they want to think there's some, combination of interventions they can do now that will massively slow down their aging. And I don't think there's really the evidence for it. And the real, I think the fundamental, to sort of boil down the challenge with Blueprint into a couple of sentences, is that it's an N=1 experiment. And he's one person, and he's doing a load of different things. And sort of the normal clinical trial way of doing stuff is to have one intervention and loads of people, so you can do the statistics easily. But if you've got one person and loads of interventions, effectively doing the whole thing backwards, it's very, very hard to unpick what's going on here. And actually, the fundamentals of the blueprint, he's doing a lot of exercise. We know that's a hugely good thing for your health. He's doing lots of, aerobic strength training. He's doing all the right stuff in that respect. When you look at his diet, he's got some very sort of narrow, specific dietary recommendations. And I think he even eats exactly the same thing every single day. But when you drill down into what those are, he's eating a lot of fruit and veg. He's eating a lot of nuts. He's getting a lot of vegetable protein. Like, these are all things that we know are fairly robustly beneficial. So whether or not you have to have the exact sort of olive oil smoothie he has for breakfast, he's probably eating, broadly speaking, the right kinds of things. And so if he's exercising, if he's getting enough sleep, if he's, eating this relatively good diet, like, are the supplements doing anything at all? Or has he just gone from, perhaps a slightly less healthy previous state to an optimally healthy sort of basic health advice state? Then all these other things might just be having no effect at all. And it'd be much more useful if he could at least, you can do it. There's a trial design called a crossover trial, which is where you give half your people the drug and half the people the placebo. Then at half time, you sort of swap over, and the people who were getting the placebo get the real drug, and the people who were getting the real drug get the placebo. And you can see if there are any changes between those groups. It's another way to sort of divine what exactly your drug is doing and how big the effect is. And you could imagine, if you were wanting to do that N=1 study, you could do something a bit more like that kind of trial design, where you put certain combinations of the drugs on and off at certain different times. He doesn't seem to be doing that. He seems to be throwing everything on the kitchen sink and measuring everything, and it's just very, very hard to know what's actually going on in Bryan Johnson's body right now.
Yeah, I believe some of the supplements that he had included in that protocol are considered or perhaps marketed as senolytics, and these are various sort of polyphenols. So perhaps we can zoom in a little bit on that. This class of therapeutics called senolytics seems to be, there's a lot of buzz around it at the moment. So what are senolytics, and why are they something that the aging field is excited by?
Yeah, it's a great question. I'm going to zoom absolutely right out to give you an answer for this one. I know that you and Matt talked about this idea of the hallmarks of the aging process. And these are something I talk about in my book. I've got 10. There are, people will give you different numbers, and there's actually been a new paper just released which would bring the total to 12. I think some of those are incorporated in some of mine, but it depends on the day of the week, exactly what's included. But what are these hallmarks fundamentally? They are changes that happen inside our biology, inside the biology of other animals. And first of all, they're things that change with age. Obviously, something can't be, causing the aging process if it doesn't change with age. And they're things that if we in the lab increase the rate of that change, we can increase the rate of aging, we can reduce lifespan, we can increase disease. And if we decrease that rate of change, it seems to slow down what we broadly define as the aging process. And that's how something gets classified as a hallmark. So they're sort of potential causes of aging. Because they change with age and because changing them changes aging, it seems as though they could be causal. We don't know yet because we don't know exactly how these things all link together, but that's the idea. And there are a variety of these different hallmarks. They start at the very, very sort of tiny level, the molecules, things like changes in our DNA. So we can get DNA damage, we can get mutations happening at the very smallest level inside cells. And then as we sort of magnify up, we can go all the way up to changes in whole systems in our body. So things like the immune system, dysfunction of the immune system. So we get less good at fighting disease. Our immune system gets less good at rooting out cancer cells, and so on and so on. And that gets worse and worse as we age as well. And in between these two scales, slap in the middle of the hallmarks, I put them, the accumulation of what are called senescent cells. And these are cells that were actually first discovered way back in the 1960s by a guy called Leonard Hayflick. And he was watching cells called fibroblasts that are just dividing in a dish. And he noticed that if he let them divide and divide and divide, then after about 50 cell divisions, something very strange happened. So the first thing is that the cells just stop dividing. But the second thing is, and I am by no means a cell biologist, by no means a microscopist, but you can see that there is something wrong with these cells. He called it the fried egg phenotype, because these cells go from these sort of lovely, sort of ordered looking roundish objects to this splat. They've got this really weird exterior shape. You can just see there's something very, very strange going on with these cells. And so they stop dividing and entered this strange state. He called it cellular senescence. We've already met that word senescence because it's just a biological term for old. So he thought these cells were just becoming old, and that's something that's going on certainly in cells in a dish. The question is, does accumulation of these cells in actual living, mice and actual living people drive the aging process itself? And it took us a very long time to get to the point where we could answer that question, but I think it was convincingly starting to be answered in the 2000s. We noticed that these cells do indeed accumulate in humans, in mice, and so on. And there are a variety of different reasons a cell can become senescent. So the crucial thing about this senescent state is it's non-dividing. And so the first reason that a cell can become senescent is that it gets a lot of damage or mutations in its DNA. Now, what are mutations? They're mistakes in the genetic code, essentially. And if a cell accumulates the wrong combination of mutations, that's how cancer starts. So if a cell's got this combination of mutations, it can learn to divide and divide and divide indefinitely, and that becomes a tumor, and that can obviously go on to kill you. So our bodies have got a number of mechanisms to try and stop that from happening, and one of them is cellular senescence. So if there's a load of damage to the DNA, the cell's looking a bit fishy, then the body just slams on the brakes, puts it into this senescent state. You can also get cells that are senescent because they've divided too many times. We've already talked about that because of the Leonard Hayflick's experiments. That happens in our bodies too. And finally, you can just get cells that are stressed. They're in a sort of stressful environment. I don't mean stressed in sort of the day-to-day, my job's stressful sense. I mean that in the molecular biology sense of stress. So there's stressful chemicals essentially going on, inside the body. So cells can enter this senescent state for a variety of reasons. And when we're young, this is happening all the time. You and me have got plenty of senescent cells in us right now as we have this conversation. But because we are relatively young, I guess, then what's going on is that these cells enter the senescent state and they start spitting out this variety of different molecules. And what the molecules are doing is they're saying, hey, over here, I'm senescent. You might want to come and clear me up. And the place that they're signaling, of course, is the immune system. So an immune cell will sense this chemical cocktail. It'll dash over to the senescent cell. It'll gobble it up. And that's just a way of getting rid of that senescent cell. And so once that senescent cell is gone, it's not a problem anymore. And that whole process can continue. So all the time in our bodies, as we're young, those senescent cells are being produced and the immune system is clearing them up. But the problem is, as you get older, these processes, they change fundamentally. So you can start accumulating more of these cells. There are more cells with DNA damage. There are more cells with mutations. There are more cells that are in a stressful environment. There are more cells that have divided too many times because that's what they do during the course of their cellular lives. But also, the immune system is getting less effective. I already mentioned that's another of the hallmarks of aging. And so it's getting less good at responding to these signals and less good at clearing up those damaged cells. And what then happens is that this it's called the SASP, the senescence associated secretory phenotype, which is a hell of a mouthful. Senescence associated is associated with these cells. Secretory, they secrete them, so they sort of give out these chemicals. And phenotype is just biological word for thing that happens. So biologists do like to give things sometimes slightly overcomplicated names. But this SASP it's essentially the hey, over here, come and clear me up immune system signal. But unfortunately, when these cells start to accumulate, that can essentially become something that accelerates the whole aging process. It can drive this process called chronic inflammation, which is a sort of hyperactivity of the immune system that contributes to aging. It can make cells become cancerous, ironically. So something that started out as an anti-cancer mechanism can actually cause cancer in sufficient quantities. And finally, it can actually even induce senescence in surrounding cells. So it's got this sort of snowballing type effect that means that it can get worse and worse and worse with time. And so this does seem to be a very obvious candidate for a cause of aging. It's something that accumulates in our bodies as we get older. It seems to drive a whole range of age-related diseases. So that might be quite a depressing story, but there's some really good, exciting news at the end of this story. And the reason we can be so sure that these things do cause age-related diseases is because scientists have come up with drugs that can remove these senescent cells while leaving the rest of the cells of the body intact. And I think the most compelling evidence of this is a paper that came out in 2018. Scientists gave these drugs to mice that were, I think, 24 months old, so that's sort of 60, 70 years old in human years. And they found that they basically, I'm happy to say, made the mice biologically younger. So they removed the senescent cells, that's a good thing. They find the mice live longer, but they're not just staggering along, with their frailty extended somehow at the end of their lives. They get less cancer, they get less heart disease, they get fewer cataracts clouding the lenses of their eyes. They can run further and faster on the tiny mouse-sized treadmills they use in these experiments. They seem to reverse some of the cognitive decline that comes with aging. So if you put a young mouse in a maze, it's often very excited to explore its new environment, find the food, whatever. But an older mouse might be a bit more anxious. Maybe it's just a bit less physically active, and so it doesn't want to go and explore. But by removing some of these senescent cells, you can rejuvenate some of that youthful curiosity. And finally, it's really worth, if listeners haven't seen a picture of these mice, again, I'm not a mouse biologist, I don't play with mice in the lab regularly, but nonetheless, you don't have to be an expert. You can see the mice that have had the drugs, they look fantastic. They've got much better fur, they've got thicker fur, they've got less grey fur, they've got plumper skin, they're less fat than the mice that have not been given the drugs. So it seems to have really this absolutely almost global effect on the aging process. And that's hugely exciting because I think this is the paradigm that we want to, have going forward, is finding a hallmark of aging, identifying a way to change that hallmark of aging back to a more youthful state, and then potentially preventing a whole range of different diseases. And so now we're at the stage where these things definitely work in mice. You've got really, really strong evidence that senolytics have an effect in mice. And so the next step is human trials. And we've already talked about this process in the sense that these senolytics aren't being trialed. We aren't just giving them to 50-year-olds who've accumulated senescent cells throughout their lives and, preventative for an aging indication. We're now using these drugs for people who've got very serious diseases. So there's a disease called lung fibrosis, which is very common in older people, and senescent cells are often found hanging around the site of this fibrotic, so effectively scarred lung tissue. And so the idea is that by giving people who've got this disease that doesn't really have any great treatments at the moment, this sort of ray of hope with the senolytics, hopefully we can prove that they work, we can see that the senescent cells get removed. And the thing I really hope is not only that obviously the drugs work, the people get better, but also that the side effects are relatively mild. Because what that means is if there's a good side effect profile, we can then say, okay, these drugs don't have many side effects. Let's try giving them to people with, late stage heart disease. That's another thing we know that senescent cells are related to, but it's something we already have drugs for. We already have diet, we already have a variety of ways we can help people who've already had a heart attack, for example. And then again, if it's safe in that population, we can spread it wider and wider still. So this is a super exciting thing because hopefully we'll know the answer to the more severe diseases within the next few years. And if we can carry on widening that net, it's probably not going to be, more than 5, 10, 15 years before we can start thinking about using these things preventatively for aging.
And when you say drugs, we're talking here about senolytics. And do these senolytics or compounds, are these typically, or are they always polyphenols like fisetin and quercetin, or are these just certain types of senolytics and it's a much broader sort of category?
It's a really interesting field, actually. So the way that the reason quercetin comes up so much is because in this 2018 paper, actually, but also in a lot of other papers from that group, in the original paper where they identified the first senolytic drugs, the way that they worked this out was they got a whole bunch of things that they thought might have some effect on senescent cells. And then they just got a bunch of cells in a dish and dripped various combinations of these drugs onto the cells in the dish. And they found the drug that killed the senescent cells, but left the bystander cells relatively unaffected. And in the end, after having tried a whole load of not just individual drugs, but as I said, combinations, they found that the most effective combination for killing senescent cells was quercetin, which is a flavonol. It's found in various fruit and veg, but it's also plus dasatinib, which is a chemotherapy drug. And it's important to say this is at much, much lower doses than you'd normally use it in cancer. But they found that this combination somehow got in there and effectively convinced those senescent cells to kill themselves, convinced them to go back onto the sort of cell suicide pathway where we'd very much hope they should have stayed. So I think some of the sort of hype around quercetin has come because it was found in that context. The real question that we don't know the answer to is at what dose and in what context. Does quercetin on its own at a particular dose have the same effect in humans that it plus dasatinib had in mice? And I think that's a really big question. There are some other flavonols that are being investigated as well. There are other actual drugs. But I think the sort of long term of this field is going to be finding much, much more powerful and specific senolytics. The reason that dasatinib and quercetin end up being used is they're effectively off-the-shelf compounds. So these scientists were trying a bunch of stuff that they had access to in the lab. But hopefully now we understand that senescent cells are important and that senolysis does seem to have positive effects, then we're going to go out and find specific drugs designed to be senolytics rather than just some flavonols that seem to happen to have that effect. And so maybe, that's an argument for eating certain kinds of fruit that have the quercetin in it. It could possibly be the case that quercetin supplements are useful. But I think the more exciting prospect is a little bit further into the future when we come up with drugs and other interventions that can kill the senescent cells. And it's worth saying these things might not even be drugs. They might not even be supplements. There are some people who come up with a vaccine for senescent cells. So you effectively teach the immune system to go after these cells a bit more effectively in older age. There are also, there are peptides. There are just a variety of different compounds and approaches. There are probably going to be gene therapies again to kill these senescent cells. So it's a really, really fast moving field. And I'd be very surprised if it turns out that these flavonols are, even in the top 10 when the dust settles and we've got the best treatments.
So is it worth taking any senolytic supplements now? I know there's a few brands that are kind of selling them at the moment or claiming that they have ingredients that have sort of senolytic properties. Is that kind of worth the cost at the moment, or are we better off just eating a healthy diet?
I think we just don't know. I think the way that I sometimes look at these things is, you find some foods that have got quercetin in that you quite like anyway and try and maybe amp that up slightly in your diet. It might not be a clinically relevant dose. That's often the case with these things. Like we know about resveratrol. If you eat grapes or drink wine, you have to eat a ton of grapes and drink 25 gallons of wine a day in order to possibly get anywhere near something that might be a clinical dose. But I think that really, we just haven't got the evidence of these things, and it's frustrating. What we need to see is these randomized controlled trials, or at least some kind of trial in humans. And one of the big challenges is that we're not even very good at counting senescent cells yet. We don't have a really firmly agreed upon definition. Some scientists say it's due to expression of certain genes. Some other scientists might use something that looks for a particular protein in the cells, and so on and so on. But there isn't even a firm agreement on what exactly constitutes a senescent cell, which means if you want to do a shorter term trial where rather than looking at lifespan, which obviously takes ages, you instead look, does this thing remove the senescent cells? You're going to find a room full of scientists arguing about what exactly constitutes a senescent cell. So I think luckily we're in a place. This is hugely exciting. There are startups. There's big money going into some of this stuff. Unless you're already very old and willing to take a risk on these things, I just watch and wait, because the evidence is going to be coming in very, very soon.
Eat your apples in the meantime.
Damn right. Keeps the doctor away. That's what I've heard.
Quick one. If you'd like some inspiration to take your fermented food game and gut health to the next level, be sure to check out my digital plant-based ferments guide. Inside are some of my favorite recipes, including my soy labneh and homemade kombucha. Learn more at theproof.com/ferments. That's theproof.com/ferments. I want to talk about Yamanaka factors. I found this part of your book super interesting, and Yamanaka factors is something that I have come across before when reading papers. I've heard people speak about it, but I really didn't fully appreciate what they were until I'd read that part of your books that was, I guess, focused on cellular reprogramming and stem cell exhaustion. So what are Yamanaka factors and what is cellular reprogramming?
This is properly fascinating stuff. And I think this is one of the most exciting, but also one of the most sort of speculative and bizarre parts of aging biology at the moment. So we have to rewind back to 2007, when a scientist called Yamanaka, as you'd expect from the name of the factors, he and his lab were messing around trying to understand what makes something called a pluripotent stem cell. So a pluripotent cell is a cell that can divide into any kind of other cell. And if you think, right back to the very beginning of our lives, of everybody listening to this lives, obviously, we started out as single cells. We started out as a fusion of an egg and a sperm from our mum and our dad. And that first cell, those first cells in the very earliest part of the developing fetus were what are called pluripotent. So these are cells that then go on to, our liver, our bones, our brains, our eyes, our muscles. Every part of your body started out as this one kind of cell, but they're able to turn into anything at all. And so for obvious reasons, that is a super exciting, cell to find biologically speaking. And so we know that you can find them in very early embryos, but how can we make these things in the lab? Is there any way that we can, come up with a way to make more of these to do experiments on them? And so what Yamanaka was trying to do is come up with an understanding of what makes a cell pluripotent. And so they identified a bunch of genes that are expressed in these pluripotent stem cells. And they were trying to work out, can we make a normal cell, so, adult skin cell or an adult liver cell or whatever it might be, into one of these pluripotent cells. And they tried this huge number of different combinations. And it's important to emphasize, like in the 2000s, gene editing was not as advanced as it was now. This is an absolutely epic feat to try a bunch of combinations of 24 genes. But they eventually did manage it. And they narrowed it down to just 4 genes, which are the so-called Yamanaka factors. And if you add an extra copy of these genes into a cell, it seems to run the clock backwards effectively. You can take an adult skin cell and it'll go back and back and back and back and turn into one of these pluripotent stem cells, which is this absolutely incredible discovery, won him a Nobel Prize a couple of years later. Really super exciting. I think a lot of people were excited about this for the sort of stem cell implications. So the therapeutic dream of this was that you could, say you had a problem with your liver, a scientist could take one of your skin cells, differentiate it back into a pluripotent stem cell, and then do the same process but in reverse and turn it into a liver cell for you. So take a skin cell, turn it into a liver cell, and we could use that to create, perhaps a transplanted liver, but one that was perfectly matched to your cells, to your DNA. There'll be no risk of immune rejection and so on. But also from a scientific point of view, just super exciting to be able to experiment with these cells. Now, all that is already incredibly cool. But what was then thought was that, what's going on in this resetting process, in the resetting to pluripotency? And what does that have to do with the aging process? Because there's another remarkable thing, another sort of proof in principle that aging is reversible, is that all of us, if we have kids, those kids are born at age 0. They're born biologically young. And I know that sounds trivial, but it is worth thinking about. It doesn't matter how old the mum or the dad is. Obviously, you can have more difficulties conceiving, and there are risks of genetic problems and stuff if, either parent is particularly old. But if you got, a 30-year-old mum, a 30-year-old dad, 35-year-old mum, a 35-year-old dad, they have kids that have the same life expectancy as a 16-year-old mum and dad do. And so clearly there's some kind of way that our biology has to reset us back to age equals 0. And so scientists started messing around with these Yamanaka factors, trying to work out what was happening to the biological age of these cells, as well as this ability to turn into any kind of cell that they wanted. And what they found was that by resetting these cells to a pluripotent state, they also reverse their biological age back to 0 as well. And it's an incredibly exciting result. And there's just no reason to necessarily expect those two things to be connected, because what you can sort of turn into is one category of fact about a cell. So that, some cells have the ability to turn into multiple types of cells, some don't. There's also biological age, which could be an entirely separate thing. But it's hugely, hugely fortunate that it seems that those two things are connected. So fast forward to the 2010s, people are starting to put these genes into mice and see what happens. And the problem is that if you put these genes into mice and just leave them on all the time effectively, it's very, very bad news for the mice. And the reason is that if, say you've got a pluripotent cell in your body, it's incredibly powerful in the sense that it can turn into any kind of cell, but it is a hopeless liver cell or lung cell or muscle cell. It's not doing the things that those cells are supposed to do. And therefore, you're going to get, if you start inducing this pluripotency throughout the body, you're going to get massive organ failure. So it's really, really bad news to turn these things on all the time. However, an experiment a few years later tried, they coupled the genes. So they had these OKSM genes, the Yamanaka factors. They coupled them to something that meant they were only actually turned on when a particular drug was given to the mice. And that means that you can give the drug all the time and have the genes on all the time, or you can only give the drug occasionally and have the genes on temporarily. And so they effectively gave the mice this drug at weekends. So it was 2 days out of every 7. They had 2 days on, 5 days off. And what they found was they actually did this in mice that age more rapidly than normal mice. They have a genetic problem so that they age more quickly. And they found that by giving these Yamanaka factors transiently rather than on all the time, they did seem to be able to improve the mouse health. And what's really incredible is we've then taken cells out of taking just cells in a dish, and you look at when you induce these Yamanaka factors, it seems that the de-aging part of the process, so the part where the biological clock is turned back, actually happens before the part of the process where the cell becomes pluripotent and able to turn into any kind of cell. And that is just so lucky, because again, we wouldn't expect these processes necessarily to be connected at all. We certainly wouldn't necessarily expect the de-aging bit to happen first. But that is so fortunate, because if the pluripotency bit happened first, you wouldn't get any age reversal, you'd just get dead mice. But in this case, you can turn it on, you can reverse the aging of the mice a little bit, and you can stop it before the cells start to change their state into something that means they're not doing their jobs. This is an incredible result, right? And so now we've got actually more experiments. We've shown that you can actually extend the lifespan of normal mice a little bit by giving them these Yamanaka factors. There are these incredible results. In fact, one came out this morning that you sent me before we started chatting, where they managed to use these Yamanaka factors induced transiently to improve the vision of monkeys that have been given an injury in the back of their eye. And by turning on these Yamanaka factors, you can reverse the cells back. And it's worth thinking about why this particular idea works. It's because if you were to injure an embryo or a fetus in the womb, then what happens is that wound can heal perfectly with no scar, because the cells are still in this very plastic state. They're still able to become any kind of cell that they want. Whereas if you do that same injury, and particularly the optic nerve, which is where they do this injury in the experiments, that's something that as an adult you basically can't regenerate. If you get damage to your optic nerve, that damage is permanent. But by turning the clock back in these cells, they probably made them a bit more plastic and made them able to do that healing process. So that's an incredibly exciting result. The real challenge is that this study in the monkeys, there was a previous study in mice that showed very much the same thing. It's done with these additional copies of the gene that are inducible with the drug. And so the idea is that you give them the drug, and this extra gene that's in their DNA that wouldn't normally be there is then able to turn on the Yamanaka factors and start the process. And what I often say when talking about this as an idea for a therapy is it feels like a piece of technology that's fallen through a wormhole from the future. And what do I mean by that? Well, it's a super powerful, it's effectively like a cheat code for our biology, right? You can just induce these 4 genes and you can turn back the clock, which is absolutely incredible. The real question is, can we, with our puny 2020s biotechnology, turn this into something that we can actually produce as, some kind of intervention for human beings? Can we, come up with a gene therapy that does this? Can we come up with some kind of drug or combination of drugs that's able to turn on these factors in the right places in the body to come up with some kind of biological rejuvenation? And the good news is, because this is so exciting, because this is so hot, I think there might even be enough investment to find out. So the really big news, I think last year, was that Jeff Bezos, the founder of Amazon, and a bunch of other extremely rich dudes had put together a huge amount of money, $3 billion, to create a startup called Altos Labs, with the mission of working out whether this was possible. I think it's a bit weird to call it a startup when it's got quite that much funding, but I think that probably is enough money to have a really serious crack at turning this, hugely exciting prospect into something that might work as a therapy. But the question is, can we do it? I'm really excited to find out, because the potential is absolutely enormous.
With regards to that potential, so if I'm following you correctly, we're talking about drugs that will turn on one or more of these Yamanaka factors in a kind of therapeutic sense, or clinically. Are we talking about such drugs preventing or reducing someone's risk of like cardiovascular disease, cancers? How do you see these types of drugs being used, or is it going to be for very specific things like vision in the case of these studies out of David Sinclair's lab?
Yeah, I think the first thing that's going to happen is they're going to be used for these regenerative treatments, probably, because again, this is, say you've got some kind of issue with your spinal cord. We don't have any treatments for that right now. And so if we can come up with some way to induce regeneration in tissues that are normally unable to regenerate, that is already super exciting. But I think the ultimate end game, and this is really a recurring story. Once you've understood this story for senolytics, you can copy paste it across every one of the hallmarks. These reprogramming factors, what they seem to do, they reduce something called the epigenetic age. So people might have heard of the epigenetic clock, which is one measure of biological aging, and epigenetic changes are another of the hallmarks of aging. It also seems that they affect other parts of the cell. So another thing that we've observed is that the mitochondria, you might have heard of the powerhouses of the cell, these tiny little things inside all of our cells that generate much of our cellular energy. Those things seem to be rejuvenated by this reprogramming process as well. We're not entirely sure what the mechanism is for that. But again, the idea would be, by intervening with these Yamanaka factors earlier in life, we can potentially prevent many, perhaps even all age-related diseases, or partially prevent some of these things by reverting your cells to a younger phenotype, as it were. So there's me using that complicated biological word again. By reverting our cells to a sort of younger state is the thing that I should say. I think to temper the excitement around this reprogramming, I think it's unlikely to be the whole picture. Because although it is super cool and we can literally reverse aging within individual cells, I think there are some other aspects of the aging process that we're still going to need to address. And I think that's really important. Although, when I say, oh, this is cool, it's got $3 billion, that, you know, they've got enough funding to find out if this thing's going to work or not. It would be a huge shame if we absolutely nail reprogramming and can reverse the age of all the cells in your body, but then you die because of something that's happening outside of your cells. So for example, we know that the proteins that are between cells, called the extracellular matrix, that can change as you age. And there's no particular reason to expect that this reprogramming will do anything to cell proteins that are outside cells. It might be because the cells are biologically younger, they start to sort of rebuild that extracellular matrix. That'd be fantastic news, and I very much hope that happens, but it might not happen. And the other thing is that the main change that this does is this epigenetic reversal. Epigenetics is so called because it's sort of above our genetics. Epi, I think is a bit of Greek. And so what that means is that it's happening, it's sort of decorating all over our DNA. But that means that if the DNA contains a mutation, if there's effectively a spelling mistake in that DNA, that doesn't get corrected by this epigenetic clock. And so although there's some evidence that it can sort of paper over these genetic mutations, it's not actually going to solve them. And so again, it'll be a huge shame if we nail, everyone's got absolutely perfect, 25-year-old epigenomes, we're all walking around feeling fantastic about our DNA methylation. But unfortunately, we've got a bunch of mutations that we haven't bothered to fix. So I think we just really need to make sure that we hit all of these hallmarks of aging. Not necessarily because we're going to need to hit all of them. It might turn out that, some combination of reprogramming and senolytics and, some third therapy is a huge, huge deal without needing to address any of the others. Or it might turn out they're connected in such a way that you can, address some subset of them. But I think we really need to make sure we have therapies for everything in order to make sure that we don't just, die of the one hallmark that we missed.
Yeah, I think you used a term in the book, smart treatments, speaking about a future. I believe what you were describing is a future where someone could sort of tap into or utilize a variety of these different innovations, senolytics, gene editing, cellular reprogramming, to tackle all of these different hallmarks of aging and live better for longer ultimately. So that's the kind of future that you see, is it? A sort of a suite of interventions that humans are utilizing to extend their lifespan?
Yeah, and I think I talk about a cure for aging in the book and in talks, and this raises the hackles of some biologists, but I'm really happy to talk about it in that way because I think that is ultimately what we should be aiming for. It is, as we said at the top of the podcast, our greatest cause of humanitarian suffering, essentially. It's a huge, huge deal. And we know that there are animals out there that are negligibly senescent, that have a risk of death that doesn't change with time. And so, will we get there in our lifetimes in the next 200 years? I don't know. But I think a cure for aging, that is what I would mean by it. And that is what, I think we should be aiming for. So that's sort of the rationale behind that. But the other problem with the phrase a cure for aging, which as I say, I do use quite a lot, so this is an issue, is that it sounds like I'm talking about a single pill. It sounds like there's going to be some magic medicine that you can just pop and it'll slow down or stop your aging process entirely. And, that's the end of that. But actually, I don't think that's going to be what happens at all. Because although, we can categorize the changes that happen with our age into these hallmarks, into these sort of 10 categories, 12 categories, whatever it might be, ultimately, these things are far more connected than I think we fully understand at the moment. And so I talk about this idea of systems biology, the idea that the hallmarks aren't actually sort of a list of, a to-do list essentially for biologists, but they're more like sort of stations on a metro map of a city with a very complicated metro system. And so we know that there are these really important stations, but we don't know how those stations are connected to each other. So to give an example, I already talked about DNA damage mutations. We know that can cause senescent cells to appear. Reducing in the length of these things called telomeres is another hallmark of aging. We know that can cause senescent cells to become senescent. We know that the immune system failing stops the immune system failing stops the clearance of these senescent cells, which is one of the reasons that they tend to accumulate as we get older. And we also know one of the reasons the immune system fails is because some of its cells become senescent. So you can see there's this really complicated sort of spaghetti of connections between these things. And so although I think we can make some really exciting progress against aging by coming up with senolytics, by developing reprogramming, by developing this suite of treatments, I think the ultimate way that we're going to cure aging is by understanding how they're all connected. And we'll come up with some really clever interventions where we can go in and sort of stabilize that whole network that sort of slowly falls off the rails as we get older and bring it back into a more youthful state. And it might be that it's some combination of the treatments that we can already conceive, or it might be that we stick all this into some massive machine learning computer model and it does some very clever stuff. And it's like, well, you want to change this gene in the lungs and this gene in the liver and this gene in the brain. And we might not have any idea how those things are actually connected to the aging process apart from in some sort of computer language way of understanding it. And it might be that is the way that we ultimately go on to cure the aging process. The way that we get there, as I was writing this, thinking this was getting toward the end of the science part of the book. I was thinking, Andrew, this isn't the end of the science part of the book. This is the beginning of the science fiction part of the book. Because we're talking about this idea of having a model for the whole of human biology, which is this fiendishly complicated thing. Talking about machine learning and AI and like all these buzzwords. And you think, Andrew, you've gone too far this time. But actually, you've got to think about what's the timescale over which we're hoping this will happen. I already said that people in middle age at the moment perhaps have 30, 40, 50 years still ahead of them for these things to happen. And we're also going to benefit from this first generation of anti-aging treatments. So, we potentially got even longer still. So let's use 50 years as a round number. Back in 1973, I think it was 1 year since the first 8-inch floppy disk had been introduced. Now, there might be some listeners who are too young to even know what a floppy disk is. It was a very rubbish way of storing an extremely small amount of data. It was the introduction of the very first 086 computer processors. So these are the sort of the very baby computer processors that have given birth to almost all of the computers that we use in the modern world. Such baby steps. And yet, now we've got these enormous computers with enormous power, enormous data storage. And we're also collecting a huge amount more data. If you think back to 1973, it was only a couple of decades since we'd understood the structure of DNA. We certainly couldn't sequence it in any great depth, in any great detail. Whereas now we've got to the point where you can get your genome sequenced for probably less than $1,000. And that means that we can collect far more data about far more people. And it's not just DNA, it's, proteins, it's proteomics. There's a whole suite of biological tools. It means we can collect so much more data. And ultimately, I think we are going to get to the point, we've already seen in the last sort of year or so, the massive advances that have come on in artificial intelligence and machine learning. We're getting to the point where we can hope that we'll, certainly in 50 years' time, be able to feed some of this huge amount of data we've collected into computers. And if we come up with treatments for those hallmarks and can start, targeting them one at a time and understand, therefore, how they're linked together, that I think ultimately is the data we're going to feed into those machine learning models, which could get us to a point where we're coming up with some, really wacky sounding treatments. We don't even necessarily fully understand how they work, but can nonetheless radically change how long that we live.
How do you think humans in, say, 200 years from now will view aging? Will it be vastly different to how we view it today?
I think that's a great question. I think it's actually a really important question because I think a lot of people see aging as this natural fact of life because we've all lived with it throughout our entire, not just our entire lives, but our entire existence as a species. And our friends have aged, our family have aged, our pets, our farm animals, all of these are animals that do experience the aging process. And so we're so inculcated with this idea that it is, a fundamental fact about being a person, about being an animal, about being an organism, that we see it as fundamental. And I think we give it a really free pass, morally speaking, because I talk in the book about this idea that if we suddenly had an infectious disease that appeared that was killing 2/3 of people, there'd be a massive research effort. There'd be a huge, society-wide effort to get rid of this thing and understand it and treat it and sort it out. Whereas as it is with aging, it's almost obscured by its own enormity. It's such a huge problem that we don't even really see it for what it is. And so I very much hope in 200 years, if not cured, we're at least a significant way towards treating aging. People are living longer, they're most importantly living healthier lives. And I think, honestly, they'll look back on us with the same degree of sort of pity and sadness that we look back on people in the 17th or 18th century dying of cholera and dysentery and all of these diseases that we now know, with the right hygiene, with antibiotics, with all kinds of different things, are preventable diseases. We've got vaccines now. We've prevented a whole swathe of problems. And, they are unabashedly understood to be a good thing. They're human progress. I think our sort of doubling of life expectancy since the 19th century is probably our greatest achievement as a species. And yet, people are really, really happy to just say, aging, it's this natural thing. Cancer is just a thing that happens. We're lucky enough to live long enough to get it. I think that the practical realization of treatments for aging is going to completely change our philosophical attitude towards it. And I really hope people are just going to realize that this is, it's an awful barbaric thing that happens to us. If I want to die, the way I want to die isn't from 20 years of gradual degeneration at the hands of multiple different diseases. I'd rather just, disappear on my sleep one night. And so I think, once we realize this isn't an essential fact of life, and that's really the sort of the gospel I'm trying to spread, then I think, it should completely change our attitude to it. And I think that in 200 years' time, people will be baffled by, how tolerant we were of this process.
Yeah, I'm with you there. Okay, so everything that we've kind of discussed up to this point is sort of the exciting science that's underway, looking at new therapeutics like senolytics and cellular reprogramming and sort of the, I guess, watch this space future of aging. What about the here and now? So starting with lifestyle, what are the things that that you personally try and do in your lifestyle, and no doubt recommend to your family and friends, to slow, sort of nurture this aging process, the big ticket items that you think have the largest impact on aging?
I think the biggest ticket items, unfortunately, are the ones that all of your listeners are already more than familiar with. So don't smoke is absolute number one. If you're smoking and you want to live a long life, this is a terrible mistake. Smoking essentially accelerates the aging process. Obviously, it hits your lungs most severely, but it increases your risk of basically all of the age-related diseases, although the lung ones are the worst. It increases your risk of cancers, of heart disease, not just lung cancer, but cancers all around the body. So do not smoke. I cannot emphasize that strongly enough. Then there's exercise. I think that, there's huge evidence to suggest that having hopefully up to sort of 30 minutes a day of fairly intense exercise is good for you, like moderate exercise for that period of time. And we also know that even the first tiny steps are super important. So if you're sedentary and you can suddenly go from that to getting a 10-minute walk a day, you're already, that's the biggest bang for your buck. You're getting more benefit than someone who's already doing 30 minutes a day of running and ups that to 45 minutes. You're really, really getting a huge amount of benefit for small amounts of extra cardiovascular exercise. I think something that's quite neglected, and I would count myself among this. I don't do as much strength training as I should, but it's strength training. And that's because we know that muscle mass decreases with age. There are a variety of aging biology reasons why that's important. Some of it's to do with things like your muscles are a great place for your glucose to get stored when you've had a meal, but also just the far more basic and everyday, the more muscle mass you have, the more stable you are on your feet. And so that means that if you reach the age of 80, a huge cause of death in older people, at least a sort of original sort of proximate cause, not the proximate cause, but the first mover cause of death, is falling over and breaking your hip. And that's often because you're not quite so stable on your feet, because you haven't got quite so much muscle mass. And that can then lead to a sort of cascade of events, which means that a lot of people who get a hip fracture die within 6 to 12 months. So again, just maintaining that muscle mass is so important. And there's a lot of evidence to suggest that, again, it doesn't matter how little you do or how late you start. I saw a really compelling paper when I was researching the book where they're given a bunch of 80-somethings a strength training program. And they found that they built muscle mass. They were able to walk further and faster after this sort of 6 weeks long program. Absolutely incredible results. And, you would not think that an 80-something, they're obviously not pumping iron, they're not lifting enormous weights, but just starting to make these first small inroads can really make a big difference. So all that stuff is sort of obvious, but I think what's really cool about it is there are two things, right, that understanding aging biology have sort of changed my perspective about those kinds of health advice. The first is we've already talked about this idea of, the longer you live, the more time you've got to benefit from these anti-aging treatments. And so suddenly I'm really concerned about making sure that I do live, those extra few years in good health in order to be, eligible for whatever treatments might become available. But secondly, I think that understanding that these things, they literally slow down the aging process. And if someone said, you can go out for a run a few times a week and that will literally slow down your aging, it's, when you're running, it's super obvious it's benefiting your heart. You can feel your heart beating, you can feel your lungs going, you understand it's benefiting your cardiovascular system. But it reduces the chance of cancer, it reduces the chance of dementia. And that's just an incredible thing that you can do for yourself. It's, it's investment in your future. And that's obviously, one of the most important pieces of health advice. So that's, why aging biology makes those sort of, what can sound, quite drab. And, we've heard them a few times before, probably bits of health advice, a bit more exciting. And then there's the stuff that understanding the aging biology gives you an insight into things that you wouldn't necessarily expect have a huge effect on how long you live. And I think perhaps the one that's most counterintuitive for me is brushing your teeth. So obviously there are a variety of good reasons to maintain good oral health, not least of which is tooth decay is very painful and can be quite expensive. But we also think it slows down the aging process writ large. And the reason is this thing that I've mentioned a few times during our chat, which is chronic inflammation. So let's rewind and see what is inflammation. Inflammation is the process by which your body responds to various threats. It's the process by which your immune system goes and does something about that threat. So if you get an infection, if you get an injury, the cells at the site of that problem will send up distress flares, very much like the ones the senescent cells give out. And they'll be like, over here, immune system, we've got a problem, come and sort it out. And in a young person, you have this process called acute inflammation. And obviously this happens in older people too, it's just less effective. And the acute inflammation happens, they rapidly go and clear up the thing, help the wound heal, blah, blah, blah. Then as soon as the problem's gone, the immune system dissipates and chills out again. And we're back in that state. But unfortunately, as we get older, our immune system does enter this sort of more paranoid state, which is this chronic inflammation, where it's sort of constantly, there's this sort of paranoia, this fizzing, buzzing of immune molecules, which accelerates the aging process in a whole variety of different ways. So we were talking originally about brushing our teeth, remember? And the reason that that's relevant is if you think about what's going on in your mouth, if you're someone with poor oral health, you've got gum disease, you've got tooth decay. The reason that dentistry is in some ways, it's quite archaic. They literally yank out teeth. They fill in your teeth with metal or plastic or ceramic. And that's because your immune system can't ultimately win that battle. Those bacteria, it's sort of war of attrition with the bacteria in your immune system. And so that is essentially a chronic form of inflammation. It's your immune response happening in this chronic fashion. And so by having better oral health, it seems that we can improve chronic inflammation and thereby slow down the aging process. And that's the sort of mechanistic understanding. This first came to light in some studies, I think in the 90s, where it was noticed that people who had cleaner teeth, better oral health, lived longer, had less heart disease than people who didn't. And the obvious thing to say to that is, well, come on, people who have better oral health, they're probably systematically different from people who have worse teeth. Maybe they're wealthier, maybe they have more time to exercise, more time to eat a better diet, whatever the disparities might be. And I think that was a very fair criticism at the time. But as you understood more about the mechanism, more about the chronic inflammation, I think it's much, much fairer to say that having bad teeth does increase your risk of heart disease. There's even some evidence that it increases your risk of dementia, because we found the bacteria responsible for gum disease in the brains of people with dementia. Now, this could be for two reasons. This could be because the bacteria are taking advantage of the diseased brain to sort of sneak in and get a foothold. But equally, it could be that the bacteria are partially driving that dementia. And I'm sufficiently convinced to, just carry on brushing my teeth, carry on flossing, carry on using incidental brushes, making sure my teeth and mouth are as clean as possible, because I think it's far more than trips to the dentist that you avoid by doing that.
That's right. Also good for the breath. Another added bonus. Quick one. A lot of people ask me for tips on buying supplements and getting blood tests. I've created zero-cost guides for both of these, which you can download from my website, theproof.com. What about food? Highly controversial topic, but when it comes to longevity, I guess three things come to mind for me at a very high level when it comes to food. It's how much food we're eating, when we're eating, and what we're eating. So if you firstly, I guess, just speak to those three buckets, would you be able to prioritize those and say, in terms of your longevity, this is the number one, that's the number two, that's the number three?
That's a good way to try and shorten what could be another podcast or two or three. I think the most important is what you eat. And I think that's the thing we've got by far the best evidence for. And I think then the sort of how much you eat and when you eat is far, far harder both to quantify, but also to stick to. And I know that you talked a bit with Matt about this in his podcast as well. The challenge is that as you get closer to humans in these animal experiments, let's rewind all the way back to the beginning of this sort of science. Back in the 1930s, it was found that if you fed rats quite significantly less, they seem to live a bit longer. And so if you feed a rat like 40-50% less, this isn't just a diet, this is like a substantial cutback in the amount of food they're consuming. But crucially, you make sure they get fewer calories, they get fewer of all the macronutrients, but you make sure that they get all of the micronutrients they need, all the vitamins, all the minerals, and so on. So it was a very carefully optimized diet. You find that these rats that are eating 40-50% less live 40-50% longer. And this is an incredible result because, again, like the senolytics, these rats aren't having the frailty extended. They seem to be living healthier for longer as well. They look better. And when they did little rat autopsies on the rats after they'd died, regardless of which group they're in, they looked very similar. So there was a similar amount of cancer in both groups, even though one of them was living half as long again, which is just this absolutely incredible result. And it sort of shocks me that we didn't then do immediately more experiments on this. But this is a field that lay fallow for sort of 30, 40, 50 years after that. But suddenly, in the sort of latter half of the 20th century, there was a rekindled interest in this. And we've tried it now in loads of different species. We tried it in yeast and worms and flies and guppies and trout and pond fleas and dogs and all kinds of different creatures. And what you seem to find is that as we get closer and closer to humans, the effect of the calorie restriction seems to get less pronounced. So, for example, a nematode, I think you can triple its lifespan by doing the best form of calorie restriction. A mouse, I think you can add about 50%. Dogs, it's more like 16%, I think, in the best experiment. It's worth saying there have been fewer experiments in these longer-lived animals as well, which might be a bit of a distortion because obviously it takes ages to do a calorie restriction experiment in a long-lived animal. But then if we go all the way to monkeys, which is the sort of closest to humans that we've got, there were these two experiments done in rhesus monkeys, which started in the late 80s and lasted for sort of 30 years after that. And I think all the monkeys are now dead, so the results are fully in, but there might be a couple still sort of hanging on. What they found was, unfortunately, the two different groups who did these two separate experiments found contradictory results. And so they found, one group found that the monkeys did live a little bit longer and a little bit healthier. Another group found that they didn't live any longer, but they did live healthier. And so there's this, we could talk for another 15, 20 minutes about the details of that if you want to, but I think the sort of key result there is that it's not entirely clear what's going on in those monkey experiments. And I don't think there's a huge appetite, no pun intended, to repeat those experiments because they're huge, expensive, lengthy trials. This is something that, as I say, went from like the late 80s to 2020 or so. And unfortunately, of course, the results weren't 100% conclusive. All we've got in humans in terms of calorie restriction studies is experiments that look at very, very short periods of time. So obviously there are groups around the world that do this for extended periods. There are religious sects and stuff that eat a substantially reduced diet and have done that for their whole lives. But often, again, they have other systematic differences with the rest of the population that makes it hard to do the comparison. So the best randomized clinical evidence that we have is, I think, for 2 years was the length of this trial called Calorie, which looked into the sort of physiological changes that happen. Obviously, they can't say anything about human lifespan in that period of time. What they could say is that these people did seem to get healthier in most ways. So their blood insulin response improved, and think typical blood tests that you do, they seem to get better.
Do you know what sort of calorie deficit those people were able to sustain over 2 years?
I think they were aiming for 25%. So they let them eat what they wanted for a week and then said, right, you're going to eat 25% less. But I think actually the compliance was more like 12% because it's just really, really hard. Like, anyone who's ever tried a diet, it's exhausting. And imagine doing that for like 2 years. Because what I was going to go on to say, the problem is the side effects. And the side effects, number one, the hunger does not go away. You might think that once you've been doing it for a year and a half, maybe your body's got used to this calorie deficit and so you're going to be in a state. But most people reported that they still felt hungry all the time, which is, no fun. There's this joke in longevity circles that calorie restriction might not make you live any longer, but it'll certainly feel like longer. And you can really see, I am not a happy person when I'm hungry. So if that was literally me 24/7, I don't think it's necessarily worth it, even if there is any lifespan benefit, which we actually don't know the answer to. And there are also other sort of more serious side effects. So a few people had to drop out of the trial because of anemia, so reduced iron in their blood. And so I ended up having to eat a bit more because of that. There's also the risk of reduced bone density that a few people were observed to have. And there's also the risk of reduced immunity. So again, it's really hard to know what this means in lifespan terms. Because if you think about the experiments that are done in mice, these mice live in this cosseted lab environment. They're really carefully kept, what's called germ-free. So they're like, free of any kind of pathogens, viruses, bacteria. But if you're a human and, you're wandering around in the world where we have flu, we have colds, we have pandemics, we have all kinds of different stuff. I don't want to be living in that world with a reduced immune function. And theoretically, I could live to 120, but I'm going to get knocked over at 80 by some dose of the flu. So it's really, really hard to know what to recommend.
Yeah, well, I mean, when I look at all of that research and then I think about personally how I want to live, I don't want to be hangry. Firstly, I want to be an enjoyable person to be around. And I want to make sure I'm a healthy body weight for sure. So I certainly think from when we're thinking about how much, we don't want to have excessive amounts of adiposity. So I think striving for a healthy body weight is important, but also coming back to what you were talking about earlier around sarcopenia and muscle, I think we're also balancing that part of the conversation when we're thinking about our quality of life and how are we able to sort of move around our environment and do the things that we want to do as we're aging.
Yeah, and I think actually to rewind to those monkey experiments, I think that really sort of seems to underline this point. There's a caveat here, which is that I sort of want this to be true. I think we're both on the same page here. We don't want to be hangry. We want to maintain our muscle mass. There are all these caveats. And therefore, yeah, you got to take this with a pinch of salt because this is the outcome that I would like to be the case. And also because, it's sometimes fun as a scientist to have a degree of cynicism and say this, trendy fad of calorie restriction doesn't really work. But with all that sort of borne in mind, the two experiments, the way that they differed, there was one done at the University of Wisconsin. And the way that was done, they decided they wanted to really tightly constrain exactly what the monkeys were eating. And so they produced these pellets for their diet, basically. There are these sort of pellets made of fat and sugar and protein and all the nutrients they needed, and everything was in really precise quantities. And there were some monkeys that were allowed to eat what they wanted, and some monkeys that were given this calorie-restricted sort of diet. They just got less of the pellets, essentially. And then at the National Institute for Aging, which was the other place this experiment was done, they thought, this isn't this isn't really how, humans don't eat pellets. Humans eat a sort of variety of different things. They tried to come up with a healthy diet that was, lots of vegetables and fish and, grains and all this variety of more normal stuff. The disadvantage, of course, is that there's no way that you can make the nutrition of that diet constant over the period of, 30 years while you're doing that experiment. So there's definitely an issue in terms of controlling that experiment in a way that's as accurate as the one that was done at Wisconsin. And what they found was the monkeys in the Wisconsin experiment, the ones that were calorie restricted, did seem to do a bit better than the ones that were eating what they wanted. Then in the National Institute on Aging, and this is the one eating the sort of fish and grains and leafy diet, they found that the difference wasn't anything like as great. And it seemed like the control monkeys eating the normal diet, quote unquote, were doing about as well as the calorie restricted monkeys eating the pellets. And then the calorie restricted monkeys eating the grain and the leaves and the fish and stuff didn't seem to be doing a great deal better than their compatriots eating what they wanted of that diet. And when you sort of look at what was in those pellets, it turns out they're quite high in fat, they're quite high in sugar, they're sort of the monkey equivalent of burgers and fries. So you can imagine if you're in the ad libitum group, the monkeys that are allowed to eat what they want, of this delicious pot of, pellets of effectively burgers and fries, and you're in, unfortunately, they aren't in the most stimulating environment necessarily, the monkeys in these experiments, you might just sit in the corner, scoffing those delicious pellets. And so maybe it's the case that if you're eating a bad diet, that is bad. We can all agree with that. And eating less of a bad diet is better than eating more of a bad diet. But then if you're already eating a basically healthy diet, then restricting that diet isn't going to do you a great deal of extra good. As I said, you've got to take all of this with a huge pinch of salt. There are other caveats in the experiment that we could talk about. But, it does seem to me, therefore, that what you eat is far more important than the quantity. And from a sort of a broader public health point of view, we already know that being obese, carrying adipose fat, carrying visceral adipose tissue inside your belly particularly, is very bad for your long-term health. And so we'd be far better off trying to help people who are, 25, 30, 35 BMI into that normal range than we would be getting people who are already in the normal range and putting them on this super restrictive, hunger-inducing diet. So I think that's definitely the thing that I took away from those experiments.
And then, of course, what we eat, the quality of our food, high fiber, water-rich, low-calorie density foods as opposed to hyper-palatable, calorie-dense, ultra-processed foods directly affects our hunger and influences how much food we eat.
Yeah, definitely. There's humans, we've got psychology, we've got physiology, there's all kinds of other complicating factors that we've got in there. I think a really good example of this, I'm not sure whether I believe this, but I sort of mention it as an idea in the book. I think it's probably, there are a variety of reasons to think that a vegetarian diet is probably better, or a more vegetable-based diet than a meat-based diet is probably better for you. And one theory is that when we talk about calorie restriction, there's a whole nother sort of kettle of fish that we've got to discuss. In what are you restricting? In the book, I call it dietary restriction because there's some ambiguity. Are you restricting calories? Is that the most important thing? Should you be restricting protein? That's something someone else, so other people talk about. We've already talked about some of the issues around that and not losing muscle mass. So that's complicated as well. Or are you restricting something else entirely? Like some people think that restricting certain amino acids, so this is constituents of the proteins, is actually what the crucial thing is. And there's one theory that the reason, or one of the reasons that a sort of more plant-based diet is better for you is because you're eating a combination of amino acids that's actually less tailored for your human biology. Because if you think about, what are you, mainly speaking, from a sort of biological cellular point of view, most of your body mass is muscle. And so therefore, if you eat a nice big steak, that is the muscle of a cow, like that is essentially going to have almost exactly the same composition as what your body needs in order to, produce cells. So that might sound like a good thing, but actually, if we believe this idea that restricting certain amino acids is one way to sort of do a bit of calorie restriction, then by eating a more plant-based diet, you might be getting a bit less of those amino acids, and so you're feeling full because you've already eaten, the whole meal's worth of plants, but you're not perhaps getting quite as many of those amino acids as the person eating the beefsteak was. And so, are you doing a bit of sort of protein or amino acid restriction by the back door? As I say, I'm not sure that I believe that is the reason, or a reason that people who have plant-based diets live longer, but you can just see the sheer complexity of trying to translate these findings that sort of happen. They've given some amino acids to some animals in the lab, then trying to turn that into a human dietary recommendation is a minefield.
On the way over here today, I had to chuckle. So I'm in Indonesia at the moment and getting around on a sort of moped scooter. And the air quality here it's a little different to Sydney, where I am a lot of time, and no disrespect at all. It's just the culture here, and there's a lot of bikes and trucks, and I had this big puff of black smoke coming at me on the way here. And it got me thinking about pollution and particulate matter and whether there is any data out there that looks at the effect of air quality, the air that we're breathing, and our health span or lifespan.
I think actually this comes down to being very similar to smoking. And obviously it's not as bad as smoking, because when you smoke, you are directly inhaling like the fumes from a burning thing. But, if you're in an environment where there's a lot of air pollution, there's quite good epidemiological evidence that bouts of particularly heavy air pollution can cause things like heart attacks and strokes to spike in the short period thereafter. And that general exposure to air pollution, particularly when you're young, can have huge effects on your respiratory and your cardiovascular health going through your life. And I would suspect there's probably also, because what a lot of this often boils down to is chronic inflammation. You'll hear that phrase over and over again if you start talking about aging biology. Probably it's causing inflammation in the lungs, and so that's going to spread out throughout the body. It might even increase risk of cancer and dementia and all kinds of different things. The epidemiological evidence is only just starting to come in. But I think this is hugely important. I've seen some calculations done where it's been worked out that the kinds of things you do to sort out climate change are reduce the number of cars burning fossil fuels, get rid of coal-fired power stations and that sort of thing. And if you run the numbers, you don't even have to think about the carbon dioxide that these things are emitting and the fact that they're warming the planet in order to make this thing economically beneficial. Like the number of lives and healthy life years you would save by reducing the level of air pollution is already economic argument enough, and frankly, human argument enough to do something about these sources of pollution. So I think air quality is a hugely important thing. And it's something that we just don't think enough about, because unless you are in, Indonesia, or I've been to Beijing and breathe that thick sort of yellow smoggy air, in sort of richer countries, in richer cities, the air pollution isn't as visible. It's not like in the 1950s and 1960s in Britain, where you used to get these huge sort of smoggy days in London. So now the air pollution is a much more insidious threat, and it probably is taking, I don't know, it's very hard to put numbers on these things, but it could well be taking a few months off the life expectancy of people in richer countries. But because the air most days is relatively clean, it's just not something that we think about. And I think that governments really need to start, enforcing the standards that in a lot of cases do already exist, but just aren't being stuck to, because it's a huge public health problem, especially in poorer countries, but in rich countries too.
You mentioned smoking, and certainly in Indonesia, I've noticed there's a lot more smoking. So in Western countries, thankfully, the alarm was rung and less people are smoking, which is great. But vaping has kind of entered as a new trend. I'm wondering if you've looked at that. I have some friends who enjoy the old vape, and just observing them and how often they're vaping, I feel like they are actually smoking more now with their vape, because it is so much more convenient and they can do it in more places than they were before they made the switch and were smoking traditional cigarettes.
That's a really interesting point. It's another place where sort of psychology and sociology start to impact on the lab studies. I haven't done a deep dive into vaping, so I won't claim to be a huge expert, but I think it's fairly clear, like just as an opening guess, that it's going to be less dangerous than smoking, because I think there are hundreds, maybe thousands of carcinogenic chemicals in cigarette smoke, whereas a vape, it's basically nicotine, water, and there are a few other chemicals that there are some question marks over, but it's nothing like the same as tobacco smoke. And also it's not being raised to anything like the same temperature as that tobacco is being raised to when it's burned. So it seems fairly likely on the sort of balance of the evidence that it will be less dangerous. The real problem is that obviously vapes haven't existed for 30 years, so we can't do the sort of big epidemiological studies that were able to discern the effect between smoking and lung cancer and smoking and all-cause death. So there is a question mark. We don't know what it'll do. And I think it really does depend, exactly as you're sort of alluding. It depends what are you replacing and what are you replacing it with. And I think the UK has actually taken quite an enlightened approach to this, which is that they've said, we know people are smoking. We know smoking is really bad for people's health. And there have been decades and decades of campaigns encouraging people to quit. And a lot of people have quit. It's not that those have been completely unsuccessful, but quitting is still very, very hard. At the end of the day, cigarettes are addictive substances. They contain addictive substances, and often there's a social element to it as well, because you're part of a group of smokers and you go out and smoke when you're on a night out and so on and so on. So getting people just to straight up quit is very, very hard. And there might, maybe the people who are still smoking, that sort of hardcore rump, are going to be very, very tough to convince to stop smoking. But if you can convince some of those people to, instead of smoking, vape, then it seems pretty likely that maybe it's not perfect, but it's probably going to improve their health outcomes versus smoking. And so the UK is very much taking this sort of harm reduction approach, is what they call it, where they said, okay, vaping might not be perfect, but it's better than the alternative. And so we're going to regulate it in such a way as to allow that to be the case. Whereas the US, I think, has taken a far less enlightened approach from what I understand, which is basically they've gone, this is another dangerous thing, nicotine's addictive, blah, 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 and sort of taken a very hands-off approach. And now you can get these super potent vapes over there, and you're in a position where we just have, the epidemiological effects are far, far less clear. So I don't really know the answer specifically, and I'm not sure we will for years, maybe even decades, hence, to actually precisely quantify how safe or unsafe they are. But I think if you're smoking and you're struggling to quit, I would definitely rather be vaping. But then, as you say, as long as you're not vaping all the time, where previously you only had a couple of cigarettes a day. So it's a really tricky situation. And unfortunately, the best advice is the boring one, which is just don't smoke.
Is there any data that you've looked at on stress? And I'm talking about like work stress or relationship stress or finance stress and how that affects aging. And has anyone looked at, interventions like breath work or meditation and then measured, aging at a cellular level?
I think we just don't know enough about this stuff, and it's really, really hard. There have been so I think the most compelling sort of fundamental biological data is that telomere length, which is so let's rewind and explain what that is, because that's something we haven't actually managed to explain so far. Telomeres are these sort of caps on the end of your DNA, and every time a cell divides, there's actually this bizarre flaw in our biology, which is that when a cell divides, it has to replicate that DNA, but our DNA replicating enzymes can't make it all the way to the end of the string of DNA. And so what that means is, if we were to be just doing this on a normal string of DNA, we might be losing important information every time a cell divided. And there are lots of tissues in the body where cells are dividing all the time. So this would be very, very bad news. So the way that evolution has chosen to get around this problem is it's just put this huge string of repeated, effectively nonsense. It's the same 6 DNA letters over and over again. TTA, GGG, thousands of times. And when you start out as a kid, your telomeres might be 10,000, 20,000 DNA bases long, DNA letters long. And as you get older, you lose, it depends on the cell type, you lose something like 100 bases per year, and they get shorter and shorter. And eventually they get critically short, and that causes a problem. And as we've heard earlier, in fact, one of the things that can happen with critically short telomeres is that your cells can enter the senescent state. So that's one of the reasons that we age. So that sounds, again, like a really good candidate for a hallmark of aging. It's a thing that changes as we age. It seems to be causative in the aging process, perhaps. There are these epidemiological studies where they've shown it's actually not as clear a biomarker as you might hope. So it's not as though I can measure your telomeres and be like, okay, your telomeres are, 11,412 bases long. Therefore, you are, 38.5 years. The specificity, the accuracy is actually much lower than you might think. There are some 90-year-olds who have telomeres as long as some 20-year-olds. So it's really quite a scattered graph. But there is a trend downwards. And that trend does seem to be in some way causative. So if you've got short telomeres for your age, you do seem to be at greater risk of age-related disease. And I think the most illustrative and perhaps the most morbid of the studies that I've seen on this was one looking at twins. And they found that identical twins, the one with the shortest telomeres, tended to die sooner. So if you're a twin, you might not want to get yours and your twins' telomere length measured, because there's a sort of morbid indicator there. So there does appear to be this relationship. And in terms of stress, we know that people who've been exposed to very stressful childhoods or stressful situations do seem to get shorter telomeres as their lives wear on. And so it could be that there's some causal relationship with the aging process there. But as I've sort of suggested, telomere length is a really bad marker of how old you are. It's very, very noisy. It varies a lot between individuals. And it also varies quite a lot, often just from like week to week or month to month. So the most common place to measure telomeres, the easiest thing to do is just a blood test. And that means you're measuring the red blood cells in your blood don't have any DNA or don't have any normal cellular DNA. And so it's actually the white blood cells, the immune cells, that are being measured. So imagine you've just had an infection, then your immune cells might have divided a lot of times. And if they divided a lot of times to fight that infection, what are they going to have? Really short telomeres. And so it can be that your telomere length can dip very, just for a short time until those immune cells get cleared out, because, obviously you don't keep them forever after an infection. But it can be that the telomere length can change quite a lot from week to week, from month to month, depending on all kinds of different factors. So that isn't like hugely compelling evidence. I think there's almost certainly something to, stress and trying to avoid it. But the real challenge is doing any kind of, because again, the gold standard for all these things is a randomized study. And, that means you'd have to somehow stress out a bunch of people for 5 years and not stress out, sort of mollycoddle a bunch of other people for 5 years and see who lived the longest. The challenge is that if you don't do that randomized study, there are going to be differences between those two groups. So who's likely to be more stressed? It might be people who are on lower incomes and therefore, have a more stressful job and are sort of living paycheck to paycheck. And so that's a stressful thing. That might mean they have less time to exercise, less time to eat a good diet. They might have poorer sleep quality perhaps because of their stress, but also perhaps because of a whole variety of other factors. And so it's really, really hard to tease out, what's the stress and what's the bad sleep quality, or what's the stress and what's the poor diet, and so on and so on. I think the main argument that I've got for trying to, decrease your stress levels, and in this sense, just like doing whatever works, like if breath work or meditation works for you, that's fantastic. And, you should keep doing it, is that even if it doesn't extend your life, I don't want to be stressed. Like I don't like being stressed. It's stressful by definition. And so even if your life stays exactly the same length, that is something that's definitely worth trying to do. And so if there is something that's within your control that you can try and reduce the level of stress, there's definitely a chance that it'll improve how long you live or how healthily you live, but it'll definitely improve your quality of life during that time. And so I think there's not super compelling data. I'm not sure we'll ever have super compelling data because you can't do that randomized trial. But I think there's just such a strong argument for doing it anyway that it's not something to don't spend too much time stressing over it, I guess.
Yeah, I guess hard to do that kind of randomized controlled trial, but maybe not as gold standard. But you could go out to a group of people who are all in the same sort of occupational life circumstances where it's known that they're under pressure and there's a lot of stress. And if there was a way to, at baseline, measure their biological age, we haven't discussed that, whether that's a possibility. But if you could, at baseline, take a group of people who you believe are stressed and then randomize them into two different groups, one who is a placebo and one who is getting some sort of meditation or breathwork intervention, and then re-looking at biological age after the intervention.
Yeah. No, I definitely love to see that study, and that would make a lot of sense. Although, I've perhaps been a little bit more dismissive than this is fair. And there have been a few studies like that. I saw a study that suggested that meditation increased telomere length, for example. But the real challenge, often they're very small studies, which is just a shame. This is clearly something that's hugely important to a lot of people. And if we could get something that's essentially a free intervention that could improve people's quality of life, it's wild that we don't know definitively whether or not that works. So the first thing is they're often quite small, which makes it hard to generalize them necessarily. And the second problem is, as we already discussed, telomere length isn't necessarily the greatest measure of biological age. There are arguments to be had about all the measures of biological age. I think epigenetic clocks are probably better than telomere length, but what exactly are they measuring is a bit of an open question. So I definitely think I'd just like to see more of those studies. It'd be nice to know, does it reduce the stress? That's already a question it would be very useful to know the answer to, irrespective of the effect on biology. And there are a bunch of biological measurements we could do. And I think the other thing, and this is something that's overlooked in a lot of different areas of research, is that you can collect the samples and then freeze them and wait. So, we could measure the epigenetic age now, and we could measure do a bunch of blood tests that we know are indicative of health now, but we could also stick a sample in the freezer. And I think actually this is something that the TAME trial, the metformin trial we were talking about earlier, is planning on doing, because we don't know what biological age tests are going to be available in 5 or 10 or 15 or 20 years. And so if we still got access to those samples, frozen, kept safe, then we could potentially look at those things retrospectively. Because often the challenge with these things, if you're going to give someone a 6-month or year-long program of meditation, that study by definition takes 6 months to a year. So if you can get that done and, in parallel, be developing the biological age test in a completely different lab, that's a fantastic thing to do. So I think that, again, that's something that we should be thinking about more, not just in this context, but in loads of sort of biological studies.
I wanted to ask you a question about men and women. Why do you think it is that women tend to live longer than men? What is it? Is it the risks that that men tend to take, or is there a biological reason for this outside of behavior?
I think it's definitely possible that there's a biological reason as well. I think there's risk-taking behavior does matter. And you notice that if you look at the sort of mortality curves of boys, I should say perhaps boys and girls, because these things split in teenagehood. Boys and girls have very similar mortality curves during childhood. But as soon as puberty hits, boys firstly become stupider. We drink, we drive cars too fast, we do all kinds of stupid stuff like that. But also, and tragically, a huge cause of death in younger men is suicide. And quite what exactly that's linked to, whether it's societal or whether it's hormonal or what exactly it might be, I think is a bit of an open question. But clearly there are sort of social differences between boys and girls and men and women. But there are also some really compelling ideas for why there might be a difference biologically. I think one of the ones that makes a lot of sense is that you'll know from high school biology, normally then a woman has XX chromosomes. So you've got these 2 chromosomes at the end called your sex chromosomes. A woman has 2 X's and a man has an X and a Y. And that's normally how we distinguish between the biological sexes. Now, although we talk about the X and the Y, what that really doesn't emphasize enough is just how sort of rubbish the Y chromosome is. It's this tiny little thing. It's got far fewer genes than the X. It's just a completely different animal, basically. And actually, that's a really intuitive example of this, is that color blindness is much more frequent in men than it is in women. And the reason for that is that women essentially have a backup copy of all the genes on the X chromosome. So imagine you've got a defective gene for one of the color receptors in your eye. Then on your other X chromosome that you got from your, say you got the defective one from your dad, then the good one from your mum will be able to sort of fill in the blanks and make sure that you've still got fully functional color vision. But if you're a man, then you've got that defective chromosome from your dad. Sorry, it would have to be from your mum, wouldn't it? Because the Y chromosome always has to come from a male. So if you've got the defective site chromosome from your mum, you haven't got a backup on that Y chromosome. And what that means is then you end up being colorblind. And that's why it's so much more prevalent in men. Now, that's obviously a really hyper-specific example involving a single gene. But it's quite likely that a whole variety of genes that you don't have backup copies for, you might just have slightly less good genes on one of the X's. And without having sort of the alternative option, as it were, you then end up having to use that less good option. And cumulatively, that somehow affects your aging. And that might all sound quite abstract, but there is some evidence from throughout the animal kingdom that this is the case as well. So in birds, they actually have the other way around their sex chromosomes. They have W's and Z's. And the sex with the identical sex chromosomes is the men, and the sex with the different sex chromosomes is the female birds. And what they find is that male birds, potentially with this backup copy because they got the same chromosome twice, do seem to live longer in general than female birds do. So there's some evidence for that. There's also another fascinating line of biological evidence, which is the mitochondria. We've already mentioned these, the things that generate the energy in your cells. You get almost all of your mitochondria from your mum. And the reason for that is when the sperm meets the egg, its mitochondria are normally left outside. And so all of your cells develop from this egg cell. It's got a combination of your mum's and your dad's DNA, so the sort of genetic material, but all of the mitochondrial genetic material, the only part of the cell that has its own DNA, is inherited from your mum. And so imagine that you're a man, and for some reason from your mum, you've inherited the best mitochondria. They're super. They make you incredibly attractive. You're virile. You're great at catching bison or whatever it was, in prehistory. And so you're getting all the ladies. You produce hundreds and hundreds of offspring. None of those offspring are going to have your incredible mitochondrial genome. However, if there's a woman who's got an incredibly effective mitochondrial genome, she can pass that on to all of her offspring. And so it might be that there's some subtle evolutionary advantage where the mitochondria have learned to favor the female genome a little bit and learned to disfavor the male one a little bit. So there's definitely that as well. And finally, I'm going to give one further reason this is a biological hodgepodge. There's also potentially testosterone just kills us. And the reason that we think that's a possibility is that there's this evidence from populations in the past who've effectively, not to put too fine a point on it, they've had their balls cut off. So it's people like eunuchs. If we look back at the Korean eunuchs who used to be effectively part of the emperor's imperial court back in sort of medieval Korea, what they found was that those eunuchs, bizarrely, they have a family tree. And you might think, how do they have a family tree if they can't reproduce? Well, they were allowed to adopt children, which would either be eunuchs or other people from the court. And so they got this sort of family tree and this incredibly detailed set of genealogical records. And what they found was that 4 of these eunuchs, I think, lived to over the age of 100. And you've got to bear in mind, they were living, relatively privileged lives, living in the imperial palace. But the average age for emperors, I think, was in their 40s. So comparably, compared to other people living similarly privileged lives at that time, they're living much, much longer. And so it looks like perhaps, removing testosterone from the equation means that men, perhaps we're intrinsically biologically more robust, but our testicles conspire to kill us.
Right now, there seems to be a lot of talk online about hormone replacement therapy for men. And there's, in fact, clinics kind of being set up both online and also offline. And I guess testosterone being used to kind of optimize sexual function, lean mass, mood, motivation. Do you think if I guess the question here is, are you replacing it because you're low in it, or are you going above a normal reference range? Are you of the view if you are replacing it because someone was low, say someone's listening now and they're 55, 60, they've gone to their doctor said you've got low testosterone, you should try testosterone replacement therapy, and now they're taking it, but now they're thinking, oh, hang on, is that actually going to shorten my life?
Yeah, I wish we had better data on this. So unfortunately, I think the most compelling information we have is from these eunuchs, which are obviously, although it's a really fascinating case study and they do live strikingly long, it's still a bit of an unusual circumstance, shall we say. And particularly because, there might be people listening to this having the converse thought, which is something that I've thought, should I cut my testicles off in order to increase my longevity? Now, there may be quality of life effects as well. Well, we can leave those, we can park those and let people make their own mind up. But one of the big questions about this testosterone stuff is, when does the crucial effect of testosterone that decreases longevity, if indeed it does, when does that happen? And it could be that happens during puberty. So maybe, and this is a sort of very hand-waving explanation, but it could be that testosterone encourages men to throw their bodies together in a slightly slapdash way, which makes them ready for reproduction that little bit more quickly, but means they fall apart a bit more rapidly as they get older. This is actually an evolutionary theory of aging that I've sort of slightly shoddily summarized there. That's potentially something that testosterone might do. So it might be that if I cut my testicles off now, then I'd obviously lose all the quality of life side that we can discuss. perhaps better not to discuss, but I wouldn't necessarily gain anything from a longevity perspective, because it might be that whatever effect the testosterone had was during my teenage years. So I really wish someone would do a bit more experimentation, we understood a bit more, do a few more trials on testosterone in older men, because obviously it can make a big difference to quality of life, especially if you've got low testosterone. But the question is, what number should we be aiming for? Should it be lower than the normal range, just because it does seem to be a hormone that accelerates aging? And I think the same is true of things like growth hormone. It's clear that they have short-term beneficial effects. You can put on more muscle mass, which obviously we think is a good thing as we age. But the longest-lived mice that we've ever produced are mice that have a genetic defect, which means that they don't produce a growth hormone receptor. So they actually do produce the growth hormone, but they aren't able to respond to it. And so it seems that growth hormone is something that accelerates the aging process. Testosterone might fall into that same category. We don't know exactly how this applies to humans. It's really important to point out, but it's certainly something that we should be thinking about when we're giving these therapies that might provide a short-term boost in mood, might provide a short-term boost in quality of life, but what are they going to mean in the longer term, and how do we trade those two things off?
Let's finish here with the ethical implications of solving aging and potentially, I'll get you to comment on that, having more humans on Earth. I think a very sort of logical question that often arises when talking about longevity is, Andrew, what would this mean for our planet? We already have a planetary health crisis as it is. Adding more humans to the equation isn't going to help. It can't help. I know that you have a sort of unique and well thought out sort of take on this that perhaps you could share with us as a way of kind of summing up or rounding out this conversation.
Yeah, I think that's the most common question I get actually is something ethical. And very often it's, what are we going to do with all the people? We've already got this sort of population causing all kinds of impacts across the earth. And I know that you and me are very much on the same page about the impacts of on the impacts of animal agriculture, for example. Huge swathes of land. The vast majority of habitable land on this planet is now being used for agriculture, and a huge amount of it is for animal agriculture, and that's having a huge impact on the environment. There's carbon dioxide, obviously climate change. We're having an enormous impact on our planet, and wouldn't more people just make that impact worse? Well, there are a few different things to say about this. The first is that I think although this problem is often cast as overpopulation, actually I really don't like the word overpopulation. And the reason is the problem isn't the people, it's the resources that we use, and it's the land that we use, and it's all the pollution that we emit in its various forms. So the first thing is I don't really think that population is the right way to think about this. The other reason population isn't the right way to think about this is that it's a tiny number of people who actually do all of the emitting of the pollution. So to take carbon dioxide as an example, because it's one of the ones where we have the best statistics, we think that about the top 10% of people on the planet in terms of wealth emit the top 50% of carbon dioxide emissions. Whereas the bottom 50% of people on the planet emit about 10% of the carbon dioxide emissions. And so what that means is, even if we were to literally kill, and I'm not suggesting this is a policy just to be clear, but even if we were to literally kill those bottom half of people wealth-wise, we'd only make a tiny dent in emissions, and we certainly wouldn't solve climate change. So clearly, the rich world are the people who've emitted more historically. That's why we're rich. And it's also the population who are emitting the most currently. So we're going to have to do something about this. And the reason we're going to have to do something about this is because we want to get all of those people in the bottom 50% up to a sort of quote unquote Western quality of life. I want them all to enjoy the diet, the lifestyle, the choices, everything that I have. That's surely our most important moral mission for the next 100 years. And so we need to come up with a way to have that sort of, as I said, quote unquote Western quality of life, but with a much smaller footprint on the planet. So by the time you're doing that, let's imagine we can get down to net zero carbon dioxide, because that's something that a lot of people are talking about, i.e., if we do emit any carbon dioxide, we also come up with a way to capture that carbon and offset it. Then 0 times any number of people is still 0 carbon dioxide. So that means that ultimately you're not having any impact on the earth. Now, I realize that's an oversimplification because some technologies aren't going to scale indefinitely, and you're going to more people does make all of these things harder. But I think that's the first sort of way to think about this, is that it's crucial that we come up with ways to lower our emissions. And once we've done that, that'll impact on everybody on the planet. The second thing to say is that even a complete cure for aging would have far less impact on population than I think it initially sounds. Because you're initially like, even if we cured aging, it would massively reduce death. It would mean that far fewer people were dying. I already said that aging is the leading cause of death in the world, and that's only going to get bigger and bigger as the years pass. But I thought, why don't I try and get some sense for what the numbers might be. So I downloaded the numbers from the United Nations, who do these projections of population, what they think the population is going to do over the next 50, 100 years. And I said, what's the population going to be in 2050 if we cure aging in 2025? Now, just to emphasize, this is a ridiculous assumption, because this assumes not only are we going to sort all the medicine, the science, all the complicated stuff out in the next few years, we're also going to roll out these treatments globally in that same time. So that's clearly not going to happen. But let's take it as sort of I'd call it a best case scenario. If you're a population pessimist, you might call it a worst case scenario. But either way, it's an extreme scenario here. If we literally cured aging in 2025, well, let's see what happens if we don't do that first. The UN projection, we're going to go from 8 billion people today, roughly speaking, to 9.8 billion people in 2050. Under current best guess circumstances. That's what's going to happen if nothing changes in medicine. However, if we cured aging in a couple of years' time, then what we'd see instead is about 11.3 billion people. So that's compared to the 9.8 that we had before. Is that a lot or a little? Well, on the one hand, it is 2 billion more people almost. It's a significant number of extra human beings, mouths to feed, carbon dioxide to be emitted, cars to be driven, whatever it might be. However, as a percentage, it's only 16% more than we'd have in the other scenario. And I'd happily work 16% harder to, reduce my carbon footprint, reduce the amount of land that my diet uses, and so on, if that meant far less cancer, far less dementia, far less heart disease. You've really always got to concentrate on the other side of this equation here, the huge amount of suffering that's caused by the aging process. And just to sort of re-emphasize, this is my mad extreme scenario in which we cure aging in 2 years' time. Like in the far more likely scenario where life expectancies gradually increase, maybe we do one day get to the point where they're going up by 1 year per year, but they're going to be sort of more gradually increasing before that. We're going to have less than 16% size of problem to deal with. And so I just think there's absolutely no argument from a population point of view that we shouldn't try and go after what I still believe is the world's greatest cause of human suffering in order to try and alleviate the climate. And I think the best way to think about this, there are loads of ethical questions we could do, and that's another sort of 2 or 3 podcasts worth of material, but the best way, regardless of the ethical question that you get, whether it's about equality of access or whether it's about whether dictators would live forever, or there are a huge number of questions this throws up. It's to reverse those questions and imagine we were living in the year 2223 and we've cured aging. People aren't dying of cancer, of heart disease, of dementia in anything like the numbers they are today. They're living much, much longer, healthier lifespans. Would you reintroduce aging into that society in order to solve a particular problem? So, say you've got billionaires who are just accumulating wealth over lifetimes and lifetimes and lifetimes. Would you introduce a decades-long process of degeneration, suffering and death for literally everyone on the planet in order to stop those few billionaires accumulating wealth? Or would you change the tax system? These are the sort of options that you have in front of you. And when you reverse the question and realize, what you would be introducing to an ageless world, in order to try and solve some, not necessarily small, but far smaller problem than aging, then I think suddenly the answer becomes incredibly clear.
Right. It becomes incredibly clear that it would be unethical not to address aging.
Definitely.
Andrew, I've thoroughly enjoyed this conversation. I do think I'm going to have to invite you back on for a round 2 at some stage. So consider that an open invite. I certainly learned a lot. Other than getting a copy of your book, Ageless, The New Science of Getting Older Without Getting Old, where else can we direct people if they'd like to connect with you online and learn more about aging?
Well, I'm on YouTube. I'm youtube.com/Dr Andrew Steele, and I've got some videos about aging. I've got one about the blueprint that you can go and check out. There's also one about the ethics of population, which goes into a bit more depth than the answer I just gave. So those are potentially interesting. If you are interested in the ethics of aging, by the way, I've got a whole free chapter of my book available online. You can find the book at ageless.link, and you can find the free chapter at ageless.link/ethics. So that's something you can check out. I'm on Twitter. I'm at Stato, if Twitter still exists by the time you're listening to this. So obviously a bit of chaos going on there at the moment. I'm on Instagram as Andrew J. Steele. So yeah, come and check me out.
Awesome. Well, I'll put a link to all of that in the show notes. Thanks again, Andrew. There we go, friends. Thank you for showing up and the effort you're making to take better control of your health. I look forward to hanging out with you again next week for another episode.